Good morning, everyone. It's my pleasure to welcome you to the Astronaut Scholar Technical Conference, which will last today and tomorrow. And if you didn't know already, my name is Colleen Middlebrooks, and I'm the new president and CEO of the Astronaut Scholarship Foundation. And I think I've been at this for 2 weeks now and 2 days. So, little grace. We're incredibly proud of the astronaut scholars presenting today. Your innovation, dedication, and brilliance are on full display, and this conference is your platform to shine. I'd also like to take a moment to recognize and thank the many people who make this conference possible. To our scholar families and friends, thank you for supporting your scholars' journey and for joining us today. To the ASF team, directors, trustees, partners, and supporters, thank you for continuing to invest in the future of American innovation. Now, it's my pleasure to introduce two very special people. First, your co-host for today, astronaut scholar alum Ashley Monahan, a 2018 recipient and dedicated mentor and committee member. And second, ASF trustee and longtime supporter Carol Fitzgerald Tyler brings a passion for empowering young leaders that truly inspires us all. And with that, Carol, I'm going to kick it over to you to kick off this first session of the Astronaut Scholar Technical Conference. Let's do this, everybody.
They told me I had to stand on stage because I'm too short to be in the video if I'm down there. I hate that when that happens. Hi, good morning. Good morning. Oh, you got to do better than that. Good morning. Good morning. Yeah, all right. I love that. Hi, everybody. First of all, Colleen, thank you so much. And let's give Colleen another really warm welcome to the ASF as our CEO. We're so delighted to have you. And again, my co-pilot, my ride or die, Ashley. Say hi to Ashley. We might have been doing this once or twice before. Anyway, hi everybody, good morning. My name's Carol Fitzgerald Tyler. I am very proud to be an ASF Board of Trustee member. And I love doing this. I'm so honored to be able to host these incredible presentations from all of you, our incredible scholars. So I want you to sit back, relax, learn. There's lots of people online watching this. So this is your moment to teach us all about the wonders that you are discovering. So with that, I want to say hello, welcome, and let's start with Avery Love, because we love the technical conference. Good morning. Look at this, a gentleman and a tall one. Welcome, Avery.
Morning. My name is Avery Love. I am a junior double majoring in chemistry and biology at North Carolina Agricultural and Technical State University. And my presentation is on optimizing methods for measuring metabolic flux in Alzheimer's disease using LC-MS. So your body has to convert nutrients into usable energy, and it does that through metabolism, like specific cycles like the Krebs cycle, the citric acid cycle, and glycolysis. So one step in this process is branched-chain amino transferase, which breaks down branched-chain amino acids into intermediates that can be used in the Krebs cycle to convert the food we eat into energy. However, interesting about this enzyme, it's actually upregulated in Alzheimer's disease, meaning it's overexpressed. So we want to figure out what this does to the brain's metabolism and how it accelerates or possibly alleviates Alzheimer's disease. So I'm going to use MS metabolomics to study how these metabolic changes affect the brain. So just for some context in the LC-MS, it stands for liquid chromatography tandem mass spectrometry. So essentially, if you don't know, it's just a molecular analysis technique that allows you to understand what compounds are within a sample based off the molecular weight. So the MS part of this essentially does this whole process twice, so we get more so we can get more accurate data. Okay, so my first part was calibrating the MS settings to be able to do this metabolomics. So we were able to, with our control of just known metabolites, a mixture of these 8 citric acid cycle intermediates, we were able to find all of them on the mass spec setting with the correct settings. However, when we applied it to organoids, we weren't able to do that just yet because it's much more complicated. There's much more metabolites than 8 in any given cell, as well as the MSMS, which is it's a lot more finicky, so we're still working on doing that. So to actually increase or decrease expression of BCAT within a cell, we use cloning. So we use these two plasmids called POKO and POV. So POKO is going to decrease expression in the cell, and POV is going to increase expression of all the gene put inside of it. So we cut these plasmids open, and then insert our BCAT plasmids into them, and then transform these into bacteria so we can amplify the amount of DNA we have. Then, from that, we'll extract DNA. Unfortunately, I was only able to do the BCAT1 plus POKO plasmid, and then put that into a virus, and then infected human cells with them so that we can decrease the expression of BCAT1 within the cells. Then we also did that as a control with BCAT2 of just the plasmid and used transfection agent, essentially the same thing as the chemical inside of a virus. Then we went on the mass spec. So that was as far as I got to the end of the semester. But the next 2 years, this will be my job working on troubleshooting the LCMS-MS to be able to, one, analyze the organoid cells, as well as to be able to develop a better quenching method, which quenching essentially just busting open the cells to release all the metabolites so that we can actually measure the metabolites using the mass spec. And you can see here, we want the quench runoff to have no metabolites in it so we can get a more accurate depiction of the metabolism of the cells. However, you can see we do have something in the chromatogram. If we want it to look like the blank, you can see it looks more like the other 2 options. So we are going to further optimize that method to be able to get a more accurate depiction of the metabolism in the cells. And we're also going to troubleshoot the cloning to be able to get those other 3 plasmids that we want, the BCAT2 underexpression, plus the BCAT1 and 2 overexpression. Yeah. And I'd like to acknowledge everybody on the screen at ASF and you all for listening and any questions.
Thank you, Avery. We have time for a question or two. Who has a question?
I saw your hand.
What was the most difficult part of this process for you?
Mass spec. It's definitely, there's so many settings, definitely finicky. Just getting the mass spec, but we got 2 years to figure that out. Mm-hmm. Also saying the word specific. Mm-hmm. Yeah.
One more question for Avery.
So to clarify, when you're doing your LCMS studies, are you doing target analyses for each of those compounds that you showed, or are you doing non-target analyses?
We're doing non-targeted. Mm.
All right. Thank you all very much. Thank you, Avery. Okay, next we have Alexa Magstad, and she's going to talk about mutant crass. Mutant crass, is that a new band? No. It is the study of colorectal cancer metastasis. So welcome, Alexa.
Thank you. Wish I was talking about a new band. Might be more exciting, but anyway. Sorry, guys. Got to hear about this. Anyway. Hi, everyone. My name is Alexa Magstad. I'm from the University of Kansas, and I am so honored today to present my research on understanding the role of mutant KRAS, KRAS is cool too, we use a couple different terms, on colorectal cancer metastasis. Awesome. So colorectal cancer, or CRC, is the 3rd most diagnosed and 2nd most lethal cancer for men and women in the United States. And unfortunately, 1 in 24 Americans will be diagnosed with CRC in their lifetime. Now, something that's awesome is that when we catch CRC early, patients have a predicted 90% 5-year survival rate. Unfortunately, though, when colorectal cancer spreads throughout the body in a process known as metastasis, that 5-year survival drops to just 14%. So there's a clear therapeutic need to understand the underlying mechanisms of metastasis and how we can target them therapeutically. So the process implicated in metastasis that I like to study is called the epithelial to mesenchymal transition, or EMT. So throughout this presentation, the most important thing you need to know about epithelial cells is they're shaped like this. They have a nice round morphology, they form tight junctions with their neighboring cells, and they have a strong adherence to their basement membrane. This means that when they grow in a 2D cell culture dish, they grow in a nice, flat, contact-inhibited monolayer, pardon me, which means they won't grow on top of each other, and they look a lot like cobblestones. However, after cells undergo the EMT, they take on a more mesenchymal phenotype, so they undifferentiate, and instead of being nice and round, they get very migratory and very spindly, and that allows them to invade through the basement membrane and metastasize. So the protein I like to study, oh my goodness, there we go, there's the mesenchymal. The protein that I like to study is called KRAS, or Kirsten-Rat sarcoma. It's mutated in 30 to 40% of colorectal cancer cases, and unfortunately, it predicts worse survival outcomes. KRAS mutant cancers are just more aggressive. However, in 2021, Mirati Therapeutics developed this small molecule called MRTX1133 that specifically inhibits mutant KRAS and stops its action. So, with this background information, I had two main questions. One, what is the role of mutant KRAS in promoting the epithelial to mesenchymal transition? And two, can we use newly developed KRAS inhibitors to stop metastasis? So to do this, I needed a model system. So what I did, I also used a plasmid, which is a small circular piece of DNA that had a mutant KRAS G12D mutation, and I inserted it into epithelial cells called RIEs. So I just popped it onto some cells, did a little convincing. I isolated some colonies using antibiotic selective pressure, and then I expanded those colonies that appeared to have taken up the plasmid. Now, to validate that this plasmid was successfully integrated into this genome, it has a nice little epitope tag. So I was able to Western blot for that HA tag, and each time I see that HA band, I know that the plasmid was integrated into those cells. So you can appreciate from that top line in each of those clones that 48 of them express that HA tag, indicating successful plasmid integration. So, okay, the plasmid's in the cells, we know that, but what do they do? Well, a lot of things. The main thing, as you can see on the right, those are wild-type epithelial cells. They have that nice cobblestone-like morphology. Then, when they're mutated on the left, they begin to get that nice spindly mesenchymal shape. So they actually will grow on top of each other, just like cancer cells do, whereas epithelial cells won't. They don't just look like cancer cells, but they act like cancer cells too. So in a cell viability assay, I determined a 273% increase in their growth over 72 hours. Okay, thank you. Awesome. So then I popped on my inhibitor to see what would happen to the EMT. And you can see in that 500 nanomolar condition that those cells actually appear to go through a reversal of the EMT and take on a much more epithelial-like phenotype. This was confirmed by protein expression changes using Western blot, plotting for two relevant proteins in the EMT, E-cadherin and vimentin, and I showed a 95% recovery in E-cadherin and an 86% relative decrease in vimentin after putting a small molecule KRAS inhibitor on these cells. So, in conclusion, KRAS G12D mutations do promote the EMT in colorectal cancer. Specific KRAS inhibitors do have the potential to reverse these processes, and through inhibiting mutant KRAS, there's a potential in preventing metastasis in colorectal cancer. I am so incredibly thankful for my absolutely amazing mentors, my super supportive family, and all of you at the Astronaut Scholarship Foundation for this amazing opportunity to connect with you all. Thank you so much.
Thank you so much, Alexa. And we have time for one question for Alexa.
Thank you. That's super interesting that you get a reversal of the cell phenotype. Can I ask, are these cells, are you still culturing them? Are they renewing? And so like the younger generations have the restored phenotype, or is it more they're like just completely reversing? Okay, that's a great question. So the cells that we treat with the inhibitor, we just do a 24-hour or 72-hour treatment and then stop. We don't continue to grow them under the pressure of the small molecule inhibitor. I don't think long-term having that, they don't do well long-term. They're not super viable, but they don't just die like we thought they might. They do appear to have that reversal. But yeah, in future studies, doing long-term inhibitor treatment would be really interesting.
Thank you so much, Alexa.
Thank you.
I've also been notified that mutant KRAS has been booked for Glastonbury 2027. Okay, next we have and welcome Natalie Rawlings, and she's going to talk to us about amino acid signaling. Welcome, Natalie.
Hello, everybody. My name's Natalie Rawlings, and I'm a rising senior at the University of Arizona, and I'm studying biochemistry. Today, I'm going to be talking a little bit about how cells sense and respond to changes in their environment and how that gets integrated into a signaling response. But before we get into technical stuff, we're going to go to Easter Island. That is where rapamycin was discovered in soil samples in the 1960s, and this was a very interesting chemical compound that had some antifungal properties and some really interesting immunosuppressive and anti-proliferative effects. So because scientists had initially figured out that rapamycin caused changes in cell growth, they wanted to tie that to specific genetics that might be affected by this compound. And that led to the discovery of my favorite protein complex, and that's what I study, which is the target of rapamycin complex, also known as TORC1 or mTOR. So mTOR, or TORC1, you can consider as the metabolic supercomputer of the cell. And as you can see from those really complicated diagrams, it has its hands in basically everything. What it does is it inputs, or it responds to a bunch of different environmental signals, such as stress, nutrients like amino acids or glucose, and it rewires the transcriptome and the genetics of the cell to produce various changes like alterations in cell growth, changes in nucleotide synthesis, essentially everything you can think related to growth. Now, why should we care about it? I already established that TORC1 is really important for controlling a lot of processes in the cells, but we care because it is a 2 billion-year-old pathway, and we still do not know how it is regulated precisely. And it's a big problem because in virtually every human disease that has anything to do with growth dysregulation, like cancer or epilepsy, there are mutations in TORC1. And so if we understand how it's regulated, we can develop drug inhibitors that are targeted specifically for it. And I kind of have this little axolotl guy here as an interesting example of how impactful understanding TORC1 really can be. Now, axolotls are known for their ability to regenerate limbs, and so scientists explore the mechanisms behind this and essentially found out that the reason is it has special signaling within TORC1. And what they did is they basically made a chimeric protein within TORC1, and they put it into a mouse. It had some of the axolotl TORC1 in there, and the mouse was able to regrow its limb. So it's pretty amazing. That was my favorite paper. And so my work focuses a lot on how TORC1 is regulated. How do we integrate and go from the amino acids present in our environment and produce a response? And so I'm going to be sharing two of my favorite discoveries that I've made in the lab. The first one being the discovery of this protein called VSB1. This is a novel regulator that I've discovered that regulates TORC1 in specific amino acid starvation conditions. And essentially, how we figured this out was looking at what proteins actually interact with TORC1 upon different starvation conditions, and then we further probed that. And the only thing that we knew about VSB1 before this is that it was a transporter of basic amino acids. And so my first favorite discovery in the lab has been that VSB1 actually inhibits TORC1 during histidine starvation. And for purposes of simplification, if you look at the half, like the red line, there's half of the screen kind of shows that it's decreasing, and then the other half shows a strong red line. And that's basically indicating that without VSB1, you cannot turn off TORC1, which means that the cell's going to be starving, and so there's no histidine, the cell will be starving, but it's not going to be turning off TORC1, and it's going to continue and grow and die. My second favorite discovery is essentially that TORC1 can actually respond to amino acid intermediates, and namely histidine intermediates is what we discovered. And essentially, this may be a potential mechanism of action through which VSB1 is able to signal to the broader pathway. So again, just bringing it back, the importance of my discovery have been making a more targeted drug for TORC1 inhibition, which is really important in things like epilepsy or cancer, where TORC1 is overactive and causing overactivation of the cells. So yeah, that concludes the research that I've been working on for the past several years, and I'd like to shout out my lab and my mentors, and yeah, I'm so grateful to be here. Thank you to ASF, and yeah, it's so great to meet all of you.
Thank you so much, Natalie. Who has a question? Who has a question? You did such a good job. Nobody has any questions.
I have a question. Do you do you think that TORC1 could be used similar, almost used in like plant systems as a to prevent like stress responses or in other sorts of organisms to limit stress response?
It's definitely possible. I honestly haven't explored, I don't know much about plants because I've been working in a yeast lab, so it's like the most basic, eukaryotic cell. But TORC1 is really important for integrating stress response, so I definitely think it would be relevant.
I have another question. Can you tell everybody what a yeast lab smells like and how that is?
It smells a little bit like warm bread.
Yeah.
Yeah. In the warm room, we have an 80-degree entire room, and that's where we grow all of our yeast, and it smells a lot like bread in there.
Oh, yummy. Thank you. Thank you so much, Natalie. Okay, next, we would like to welcome Emily Wang, and she's going to talk to us about cancer's resistance to chemotherapy from the sweet side. Welcome.
Hi everyone, my name is Emily and I'm from Miami University. Today I'll be presenting my research, The Sweet Side of Cancer's Resistance to Chemotherapy. All cells are hungry for glucose, and so in order to transport glucose from the blood into the cell, we need these things called glucose transporter proteins, or GLUTs. There are 14 different types of GLUTs, separated into 3 main classes, and from the figure, you can see that different GLUTs are expressed in different parts of the body. Though all cells are hungry for glucose and sugar, cancers are typically much hungrier and require lots of glucose to support their rapid growth. Thus, it has been found that many GLUTs, especially GLUTs 1 through 4, are overexpressed across many cancers in order to facilitate this greater sugar intake. One major challenge in cancer treatment is when cancers become multi-drug resistant, or MDR, making chemotherapy less effective over time. The most prominent way in which cancers achieve this is by increasing efflux transporters, such as the PGP protein, which essentially takes a drug from the inside and pumps it outside so that it never actually has a chance to kill the cancer cell. So considering both of these cancer hallmarks, my main question looks at whether cancer cells become more or less hungry, perhaps through expression of more or less glucose transporters as they become more and more resistant to chemotherapy. The unique thing about my project is that I cultured cells to be resistant to a range of chemotherapy concentrations from 0 to 1,000 nanomolar. So my cell line is B16 melanoma, aka the most deadly form of skin cancer, and my chemotherapeutic is called doxorubicin, or DOX, and this is also commonly known as the red devil due to its bright red color and extreme potency. Immediately, I see that cell shape and cell size become more variable from the parent to the 1,000 nanomolar resistant cells, aka from lowest to highest resistance, confirming that changes are at least occurring on the macro level. To track whether my cells take in more or less glucose, I fed them 2-NBDG, which is a glucose molecule attached to a fluorescent tag. From these flow cytometry results, I see that the peaks are a lot closer for the 1,000 nanomolar resistant cells, indicating less glucose uptake with resistance. And this was consistent whether I added a GLUT inhibitor in the top panels or pre-starved the cells for 0 to 16 hours in the bottom panels. Looking at individual GLUT expression by Western blot, I find that their levels remain quite stable, with GLUT2 being non-existent. This is in contrast to the PGP efflux transporter, which is clearly increasing with increasing DOX resistance, as indicated by those bands becoming progressively darker. Visualization by confocal microscopy also confirmed these findings, and that GLUT expression is minimally changing, as shown in the green channels, and PGP noticeably increases in brightness. I also find similar patterns at the gene level. So many GLUT genes, formerly called SLC2A, do not change across the resistance spectrum, and as shown in both of my RT-qPCR and RNA-seq line plots. However, I did notice that the MDR genes, specifically the PGP genes ABCB1A and ABCB1B, were the only ones to noticeably spike, achieving over a 2,000-fold increase, while other major MDR genes like ABCC1 and ABCG2 in green and red remain flat and unaffected. Exploring my RNA-seq analyses further, I find that there is a broad decrease in many glycolysis enzymes, which are responsible for breaking down and converting glucose into usable energy. This can be shown by the collective dip on the line plot, the large chunk of genes from red to blue in the heat map, as well as the levels of key intermediate enzymes on the glycolysis KEGG map being blue and downregulated. With that, I'd like to conclude that as I gradually increased the amount of chemotherapy I administered, my melanoma cancer cells proportionally expressed more and more PGP efflux pumps. However, this was not accompanied by a change in glucose transporter expression. Rather, I find decreased glucose uptake and decreased glucose breakdown, suggesting a defense mechanism in which cancer cells conserve all of their energy and slow it down in order to survive chemotherapy. With that, I'd like to give special thanks to the Astronaut Foundation and all of you all for letting me present today. I would also like to greatly thank Dr. Rock Mancini and the Mancini Lab, as abundantly pictured here, as well as my collaborators from Dr. Chin Liang's lab. Thank you so much.
Thank you so much, Emily. We have time for 2 questions, so who's got the first one?
I was just curious if you kind of had any guesses as to why you didn't see expression of GLUT2 throughout any of your cells.
That's a really good question. So if we look at what GLUT2 is normally expressed in, it's normally expressed in the liver or like the kidneys, specifically to like transport glucose in and out. What I'm working with is melanoma, so it doesn't really have that function of like transporting glucose in and out of the cell. It's usually expressed in cells that are like doing gluconeogenesis or things like that. But we did see a lot of GLUT1 and GLUT3, which are commonly overexpressed in across the entire body.
One more question.
Amazing talk, Emily. Have you looked at any type of like fuel source switching or like alternative metabolic pathways that the cells are going through? Are they doing fatty acid oxidation instead?
That's a great question. I did look at whether the citric acid cycle changes or fatty acid metabolism and also amino acid metabolism, and none of those key enzymes did change, so it was only the glycolysis that actually changed.
Thank you so much. Thank you, Emily. Next, we'd like to welcome Chloe Alston, and she's going to talk about starving a virus. Welcome, Chloe.
All right, good morning, my esteemed guests and ASF family. My name is Chloe Alston. I'm a senior biology major at Howard University and a member of the PARI lab. Today, I will be presenting a project titled Starving the Virus, Iron Chelation as Adjuvant Therapy in HIV Treatment. And this work was completed as a part of my DC Center for AIDS Research Fellowship I completed this past spring. So our lab investigates how iron contributes to conditions like iron overload disorders, cancer, neurodegeneration, and what I'll focus on today, HIV replication. Specifically, I've been working on designing and characterizing small molecule chelators that can bind to iron and potentially reduce its availability to HIV. This could lead to new therapeutic strategies to complement existing antiretroviral treatments. Iron is the most abundant element by mass in the Earth's crust, and life has evolved to depend on it in numerous ways because it powers numerous body systems. But what makes iron so important and powerful is also what makes it dangerous. Its redox activity, or ability to change between different charge oxidation states very rapidly, can lead to oxidative stress and tissue damage if it is not tightly regulated. And when the body cannot regulate iron well, such as through the failure of ferroportin, one of our body's only iron exporters, that's when toxicity and infection risks go up. And this delicate balance is what makes iron both essential and potentially harmful. So HIV is a virus that rewires our immune system and has a hunger for iron. Infected immune cells, especially macrophages, often accumulate that iron. And this fuels the virus in two ways. It supports the activation of key viral enzymes, and it boosts transcription of viral RNA. But our hypothesis is very simple but very powerful. If HIV thrives on iron, can we starve it by removing iron from the system through chelation therapy? Iron chelation therapy is not new, however, and it has been used for decades in patients with iron overload due to transfusions. One of the best known chelation drugs, deferoxamine or DFO, is effective but is not ideal. This is because it's hydrophilic, has poor membrane permeability, and must be injected into the muscle, which is very painful and time-consuming, and it has a short half-life. We're now thinking of how we can redesign chelators to overcome these limitations and possibly even target HIV and starve the virus. So our goal is to make selected changes to known chelators in order to improve how they act in cells. Specifically, we want them to have greater efficacy, bind iron tightly, be more bioavailable, and not harm healthy tissues. So here, looking at our ligands, we have L1, L2, L3, and 4. Between the ligands, you can see there's very small but very mighty structural differences between the compounds. Between L1 and L2, the pyridine ring has a carbon swapped in, and so now we have a benzene ring. And then between L2 and L3 and 4, there's a CH2 group that's added that allows for that benzyl group to rotate and have extra flexibility. The L3 and L4 are also longer, and so that leads to their hydrophobic and polar properties as well. L1 and 2 have been around and tested for some time. However, L3 and 4 are the novel analogs that were designed for that flexibility and solubility, and hopefully for better medical application. All of these ligands, however, are meant to be superior to DFO, with L3 and L4 having that potentially strongest application. So our ligands are designed with their thiosemicarbazone backbones, that is the nitrogens and sulfurs you see here, and those softer electron donors are what gives it the heads up, or the greater hand compared to DFO, which has strong oxygen donors, which are very heavy and lead to problems with permeability. And so for our methods, we wanted to characterize before crystal formation and see these ligands. So we use mass spec, we use NMR, infrared spectroscopy to view the identify bonds and functional groups. And then once our crystals were formed, we use X-ray crystallography to view the 3D structures. And these combined methods gave us the full molecular picture. Then this is just a blown up image of that infrared spec of L4, so you can see those different functional groups that we confirm. In order to form crystals, we use vapor diffusion method to grow single crystals at a time. And here there's a solution of ligand along with the solvent, and then a more volatile solvent on the outside. And then over time, diffusion occurs, and we end up with a crystal. And this is how our lab did it, our iteration. You'll see we have the larger vial on the outside with the more volatile solvent, and then diffusion is occurring with the smaller vial containing our ligand. So then we wanted to see the ligands in their unbound form. So they lie in extending conformation here, and the flexibility introduced into ligands L3 and L4 can be seen in this bottom panel. And so then we wanted to see, can these ligands bind and potentially remove metal? Well, what we found is that when bound to iron, L2 has 2-to-1 binding, so that means there's 2 ligands to 1 iron ion. But when L2 is bound to cadmium, we found it bound 1-to-1, so 1 ligand to 1 ion. Then for the cadmium-bound complex with L4, we wanted to test this again, but see what's the difference between L2 and L4. This is the image of L2 and cadmium once again, and so I can show you when bound to L4, we had something completely different to what was just previously shown. There were 4 distinct complexes observed, 2 1-to-1 monomers, and 1 2-to-2 chloride bridge-mediated dimer here in the middle. But what makes this so amazing is that it's highly unusual to see all 4 complexes observed in one unit cell. And this structure shows more promise for efficient chelation, and we're excited to do future studies on it as well. This is it in the unit cell to show you that. And so in summary, we found that if HIV needs iron to thrive, then we can safely remove iron from infected cells. Then we could potentially suppress the virus without toxic effects. Iron chelators like L4 might also lower oxidative stress, which is a very big factor in disease progression. And this approach is not replacement for antiretroviral therapy, but instead could work alongside ART as adjunct treatment, especially in those iron overloaded individuals. And overall, it is just a very unique mechanism that targets viral metabolism. Our future directions include completing the iron binding studies. We'll test how these compounds suppress HIV in vivo, and then we're also thinking long-term, looking at how HIV and neurodegeneration are connected. And ultimately, we hope that this work opens new doors to antiviral and iron metabolism research. Then I would like to acknowledge my lab, my DCCFAR fellowship program, and then of course, the Howard University Department of Microbiology and the Astronaut Scholarship Foundation. Here are different publications and presentations. The number one, that's one that's in review right now, a paper in review. And then I would like to say thank you and open the floor for questions.
Thank you, Chloe. We have time for one question for Chloe.
I have a quick question. Amazing talk and research, Chloe. This is incredible. I was wondering exactly how strong these chelators are, and so for an individual who might have like more iron than normal, but not too much iron, would they risk becoming iron deficient based on how much they receive? Or like how exactly do you balance that?
I would say thank you for that question first. While we are not in the drug dosage design, what we have thought is that we know that there's liable iron that floats around in the body. A big problem is that free iron. We want to ensure there's no free iron floating. However, if an iron is in a system, we want it to stay in that system. When we were designing our different chelators, we designed them for selectivity. And so a part of that selectivity is ensuring that they target only the iron that is liable and can cause harm to your host. And that's something similar when looking at the HIV aspect of things. Like, people like to ask also, if people have HIV, could they be anemic? And then how would that chelator impact them? But it's really just we want to make sure we select just the iron that's fueling the HIV, take that out, and then allow that iron to be used in the body systems it belongs to.
Thank you so much. Okay, I had to stand up for this one. We're going to hear about wild lemurs and the insights they provide. Jocelyn Stevens, welcome.
Hi, I'm Jocelyn. I'm a biology major at the University of Kentucky. However, today I'm going to be taking you on a quick trip to the island of Madagascar to talk about what we can learn about our gut microbiome from lemurs. So the gut microbiome is a community of microorganisms that inhabits the gastrointestinal tract of all primates. And it plays really important metabolic roles in helping us digest food that we don't have the enzymes to digest, and also plays a big role in our immunity. However, despite its physiological importance, there's still a lot we don't know. For example, how it responds to the environment of its host organism, and also how it helps hosts acclimatize to ecological stressors. So I study lemurs, which are primates endemic to Madagascar, one of the most biodiverse places on the planet. And there are over 110 different species, but I study red-fronted lemurs because they inhabit tropical rainforest. And their habitat is a really great natural laboratory because it experiences a lot of ecological variation in a very short amount of time. So specifically, they experience a very hot, wet season from the months of October to April, followed by a very cool, dry season from May to September. And this also comes with dietary seasonality, so they are frugivorous, but during the dry season, they have to expand to other fallback foods like leaves and seeds. So to test whether or not this seasonal variation has an impact on their microbiome, we extract gut microbial DNA from fecal samples. For this study collected over a period of 1 year. And we found that their microbiomes are highly seasonal. So throughout the year, their gut microbiome takes on two distinct community compositions. And these compositions follow a very predictable cyclical pattern. So here we have bacterial diversity in the gut, basically just the number of species. But we can see that in orange during the dry season, there's a big jump in diversity. But as soon as the beginning of the wet season hits, we see it revert back to its previous wet season state. And when we look at the abundance of two very functionally important phyla, Bacteroidetes and Firmicutes, we see a similar cyclical pattern, but also an interesting trade-off between their abundances that's separated between seasons. And this makes sense in the context of diet because during the wet season, Bacteroidetes kind of dominate the microbiome because they are specialized in carbohydrate fermentation, which, as we know, encompasses fruit. But during the dry season, when they expand their dietary diversity, we also see an expansion of gut microbiome diversity. But also, the Bacteroidetes from before are kind of replaced with Firmicutes, and these are bacteria that are specialized in fiber and cellulose degradation, which are in leaves. So this shows that microbiome plasticity, that is the ability of the microbiome to change over time, can help buffer nutritional differences. And interestingly, all of the results from the study were nearly identical to those found in a study done by Stanford researchers on Hadza hunter-gatherers. So these are people who still consume a very seasonal, very traditional, non-industrialized diet. And so our study just adds to this, showing that our ancestral microbiome, not just of humans, but also our most distant primate relatives, are highly flexible and highly in sync with the seasons. So we conclude that the gut microbiome and gut microbiome plasticity is likely an adaptive mechanism that evolved early on to help primates deal with food stress and rapid ecological change. And I always like to end on a note about conservation, because as you might know, primates are the most endangered, or lemurs are the most endangered primates on Earth, mostly due to human disturbances. So what our lab is trying to do is leverage non-invasive sampling techniques, such as the use of fecal samples, to help monitor the health of these individuals, but also the genetic health of critically endangered populations. And I'd like to thank my mentors, my wonderful field team in Madagascar, and of course ASF for this opportunity. Thank you.
Thank you so much. Well done. Who has a question about wild lemurs?
Yeah.
Hey, I'm curious if you actually got to go to Madagascar, and B, I just was wondering about, like, what do you think the future implications of your work will be?
Yeah, I was actually just there 2 weeks ago. So yeah, I was collecting samples for like a longitudinal version of this, and we also have like 6 other studies sort of related to this. Moving forward, actually, so when we were doing this work with the microbiome, we were thinking about how we can try to get not just microbial DNA, but the actual like host genome, the lemur genome, from fecal samples, which is really hard because it's very fragmented DNA and very low coverage. So right now we're trying to figure out if we can do some genetic studies on these by extracting the actual host DNA from fecal samples.
One more question.
Great talk. Do you think like the microbiome is changing because of what they're eating, or if it's like cycling with time and has synced up with kind of the wet dry season?
So usually the microbiome, we do see changes over time, but usually it's kind of stochastic, so like pretty random. We do think that the diet does play a major role, of course, but there's also other factors that are linked to seasonality that might play a role. So like we found correlations between precipitation and temperature and microbiome diversity that, of course, can play a role in like what edible vegetation there is, but also things like precipitation, like watering hole availability. When they're like drinking water, they can passively take in other transient microbes and things like that. So those can also play a role. Also social behavior changes and reproductive seasonal changes.
Thank you so much.
Thank you.
So from the world of Madagascar, now we're going to Plants Fighting Back with Charles Colvin. Charles, welcome.
How's it going, everybody? My name is Charlie. I'm from Penn State, and I am a plant science major. So because of that, I think I have to spend a little more time giving some background as to actually what plant science is and some of the challenges that we face. Because no offense, I think a lot of you are engineers. So
I was trying to figure out how I can best explain this, and I came up with an analogy that hopefully makes some sense. So in plant science, we have a kind of overarching challenge that we're always trying to solve, and I'm going to try and put it in engineering terms. So imagine by 2050, I tell you to build an electric car, right? That's doable. You think you can do that. But what if I tell you I need to double the driving range? Okay, that's a little more ambitious, but maybe it's still possible. What if we need to use half as many batteries when we do that? That becomes even more difficult. And what if I also need this car to be twice as fast? So now I want 0 to 60 in like a second and a half. And oh, by the way, this car needs to be available, affordable, and usable anywhere in the world. So even in sub-Saharan Africa, where they may not even have paved roads, I need you to have a way with this car that can be used and charged and driven in 25 years. Good luck. So this might seem like an impossible challenge, but unfortunately, it's actually very real for us plant scientists. Because what we need to do is by 2050, we need to double our crop yields. We need to do it with half as much land, water, and fertilizer, and we need to make crops that are more nutritious. And the reason we need to do all these things is because by 2050, it's expected that we'll have a global population of 10 billion people. Now, to feed all those people, we need to increase the amount of food we produce, and unfortunately, unlike a car, there's a much more severe downside if we don't, right? We don't want people to starve to death, and so we need to find a way to fix this. There's one more problem, though. When you make a car, if you make it now and you then try and use it in 2050, the range is going to be the same. But in plant science, the climate is not the same, and so the yields of a crop now is not going to be the same as the yield of a crop in 25 years. Because we are facing increasing amounts of drought, diseases, and insect pests, and although we currently have some pesticides that we can use to combat some of these things, we're also finding increasingly that they're not very good for the environment and they're not very good for human health. So now we're facing an issue of we're trying to grow more food on less land with less resources and less inputs, and we need to do this, otherwise people are going to die. So how do we solve this seemingly impossible challenge? Well, I'm part of a genetics lab, and so what we do is, much like an engineer, we simply try and just design a better plant. And we use a variety of techniques, and we use this to target a variety of things. The example I'm going to show you today is actually focusing on the pesticides and the insects. So my lab works with corn, and there's two main caterpillars that really cause a lot of damage to corn. The one is the fall armyworm, the other one is the corn earworm. And really the only thing that you guys need to know about them is that they cause billions of dollars in damage, and they consume enough corn to feed millions of people. So obviously, we'd like to be able to control these, and like I said, we'd like to be able to do it without pesticides. So how do we do that? Well, my lab does it with purple corn. We've developed these corn varieties that are very high in a type of pigments called flavonoids. And these flavonoids are really interesting, not only because they provide these interesting colors, but also because they're toxic to caterpillars. And so what we can see is that caterpillars which feed on a higher amount of flavonoids have a much higher, or caterpillars which feed on a lower amount of flavonoids have a higher body weight, but caterpillars feeding on a higher amount of flavonoids have a lower body weight. And conversely, the mortality increases as we increase the flavonoid concentration. And that's really interesting because it holds up for both species of caterpillars as well. So we have these varieties of corn that we've developed that are high in these flavonoids, and we can see they give much higher mortality to the insects. And so now we've developed a variety of corn that we can grow out in the field, and it can be naturally resistant to these insects. And so we don't need to spray with pesticides. We can reduce the amount of inputs we need, but we can also still maintain our yields. And so this is kind of a really interesting foundation for us that we can use this technique for different challenges, not just for insects, but for other things like drought and disease. And hopefully in doing this, we can help prevent a mass starvation event that's impending. So I know a lot of you are doing very interesting research right now with things in space travel and curing cancer, but I think sometimes it's important to think about if we can't even feed the people on our own planet, how are we going to think that we can put people on others? So, and with that, I'd like to say thank you to my lab, ASF, and all the funding agencies who supported this work. Thank you.
Wow, thank you so much. When you said purple corn, I heard Purple Rain by Prince. We're in our purple era over here. Who has a question?
I have a quick question. And as an engineer, I understood that perfectly, so thank you.
Thank you.
As a plant scientist, this is coming from the engineering side, what's like the time scale of your experiments since you need your plants to grow? Like I can go in the lab and like chug out new experiments fairly fast, but I have to imagine that's a challenge for someone like you.
It depends on what we do. If we want to do things in the field setting, which is kind of the gold standard, right? We grow things out in the field, how they would actually be encountering the insects in the environment. We get one chance a year because we have one growing season, and so we get to plant once, we get one cycle a year, and if we don't get the data we want, we have to wait again for a whole other year. So sometimes that can be slower. We can sometimes supplement that with greenhouse experiments or doing experiments with younger plants, But if we want to do our best quality experiments, we get 1 shot a year.
Amazing talk. I am not in your field, but I have a question. So, correct me if I'm wrong, but my impression of corn was that, especially corn, the pesticides are often like already baked into the seed. So, how do you test this purple pigment sort of experiment on corn that potentially already has pesticides baked in? Or are you able to isolate that, or use a different species of corn that's non-GMO or something?
Okay, so there's 2 types of pesticides that can be baked into the seed. There's the ones that are genetically encoded by the plant, so that would be your GMO plants that have they call it a Bt toxin, and so that kills insects by perforating their gut membrane. So we can avoid that by simply not using GMO corn. That's easy enough. The other type is a seed coating, where it's like they put a neonicotinoid on the actual seed, and that's a pesticide. But again, if we just don't use that on our corn, we don't have to worry about that.
Okay, great.
Thank you so much. Very interesting. What a novel concept. Let's feed Earth. Just saying. Next, we would like to welcome Samuel Rosine. I hope I said that right. And he's going to talk with us about extracellular vesicles. Welcome, Samuel.
Sorry, I'm trying to figure out how to, I've never used this before, so. Green button, okay. Alrighty, thank you. Good morning, everyone. My name is Samuel Rossini. I'm a student in the Integrated Vascular Biology Laboratory at the University of Colorado Boulder. And today I'll be discussing adipocyte-derived extracellular vesicles and their role in obesity-related cardiovascular disease. So by way of some background, obesity prevalence in the US today is around 40% of adults, with projections from the National Institute of Health estimating that by 2030, half of US adults will have obesity. Obesity is associated with increased risk of cardiovascular disease, specifically ischemic stroke and myocardial infarction, but the mechanism underlying this increased risk of cardiovascular disease is not fully understood. My work focuses on studying circulating adipocyte-derived extracellular vesicles, or ADEVs. ADEVs are small membrane-bound vesicles released by adipose tissue that aid in cell-to-cell communication and the regulation of various metabolic processes. They achieve this by carrying diverse cargoes, such as microRNA, influencing the function of recipient cells, and these ADEVs are now recognized to be a key in the understanding of the pathology of obesity-related vascular disease. My previous work has demonstrated that these circulating ADEVs are elevated in adults with obesity as compared to normal weight adults. And in the coming year, I will expand upon my work by isolating these circulating ADEVs from blood samples from both populations. They will then be cultured with human coronary artery endothelial cells and human cerebral microvascular endothelial cells. The key outcome of this study will be to determine the ADEV's effects on the cellular nitric oxide, endothelin-1, oxidative stress, inflammation, and microRNA. And these outcomes are all etiological factors that are key in the development of cardiovascular disease. The findings of this research will allow me to determine if ADEVs are potential systemic mediators of obesity-related vascular disease. With regards to future research, applications and direction, with continued research, ADEVs could be a viable target for therapeutic interventions, and these findings will also contribute to research that is currently underway that aims to determine whether lifestyle interventions or pharmacological interventions may alter the production, composition, or function of these vesicles, potentially reducing the risk of obesity-related vascular disease. And in the long term, my research hopes to contribute to precision medicine approaches, which therapies are tailored to reduce the cardiovascular morbidity and mortality of obesity-related vascular disease. I'd like to give a big special thanks to the Astronaut Scholar Foundation and the sponsors for this incredible event and this incredible honor and allowing me to share some of my work with you guys. Thank you very much.
Thank you. Thank you so much. Who has a question?
Kind of curious, very fair if you don't know. Do you know what current regulatory frameworks regarding extracellular vesicle therapeutics look like, and if EVs are currently used in clinic at all versus in more of the lab stage?
So are you saying ingesting these injecting ADEVs in individuals, or? So the issue with that is because ADEVs match the physical characteristics of their parent cells, you can't just go ahead and inject them in someone because you're not fixing the source that which they're coming from. So first, to do that, you'd have to fix the source of the cell that's causing all these ADEVs to be released and causing these negative vascular effects. But it'd be beneficial to then go after you change the source of the parent cell, if that makes sense.
Thank you so much.
Yeah.
One more question.
For the future research, are you planning to use mirroring models like rats or mice, or what's your plan for the approach for that?
So we're just a human laboratory, so it'd be interesting to see what people do in other animal studies, but for our laboratory, we're just human studies.
All right, well, glad that there's humans involved. Thank you very much, Samuel. Very good.
Thank you.
In our world of AI, one begins to wonder. Okay, next we welcome Megan von Abbo, and she's going to talk with us about memory consolidation. Welcome, Megan.
Cool. Hi, everyone. My name is Megan von Abbo, and I go to Penn State. I'm a rising senior, and I work in the QUAPIS lab, where I focus on determining how the circadian rhythm influences memory formation. Today, I'll specifically talk about how hippocampal PER1 contributes to time of day effects in memory consolidation. So to start, the circadian rhythm is an intrinsic oscillation that contributes to many different biological processes across the 24-hour cycle, including, I lost it, there we go. Sleep-wake timing, feeding, and the one I'm most interested in, cognitive functioning. And it does this through zeitgebers or cue givers, with the most prominent one being light. Organisms can have a variety of different circadian patterns in regards to when they're active. So as humans, we are diurnal, meaning that we're active when the light is on and inactive when it's dark. Other animals, such as rodents, are nocturnal, meaning that they have the opposite pattern. Interestingly, though, an organism's activity pattern does not always indicate when their memory is the best. Here we have zebrafish, which are diurnal, meaning they're active during the day, and their memory is best during the day. But other organisms, like mice, are nocturnal, but their memory performance is still best during the day. And then you have other organisms, like Drosophila, which are diurnal, but their memory performance is best during the night. So we're interested in what is controlling these oscillatory memory patterns. So to start, we obviously had to ensure whether this time of day memory effect was able to be seen in our lab. So to do this, we ran the behavioral paradigm object location memory. In this task, we habituate the mice to being held and being placed in the environment that we'll be using for the testing and training conditions. We did this for 6 days in a row for 5 minutes each. Then after 24 hours, we put them in the training condition, which is the same box, the same wood chips, but they have 2 identical objects. In our case, we use beakers filled with cement, and we put them at 2 distinct locations. We then put the mice in, they get to explore, and hopefully are forming memories. Then after 24 hours, we change it to the testing location, or testing environment, where one of those objects is moved to a novel location, but they're still the same identical objects. And then we have the mice explore again. If the mice have a memory of their training, then they'll be exploring the novel location more because mice prefer novelty. And then we are able to determine whether or not they have that memory by calculating a discrimination index, which basically takes the amount of time they're exploring the novel location and comparing it to the time that they're exploring the familiar location. From that, if the DI, or the discrimination index, is high, that means that they have a memory of their training. If it's low or 0, that means they don't have a memory of their training. Then to test this, we test 6 different time points during the day-night cycle, 3 of which were during the day, which will be in yellow throughout the whole presentation, and 3 of which were during the night, which will be gray throughout the whole presentation. With this, we tested at those 6 different time points and found that we were able to get that nice oscillatory pattern with their peak memory performance at ZT5, which is around noon, and their trough memory performance at ZT17, which is around midnight. We also collapsed the data just to show in general that their memory performance is more robust during the day as compared to night, which is what we were seeing in the literature. We then wanted to determine which genes were contributing to this that were being differentially expressed and contributing to that oscillatory memory pattern. So we ran the same experiment but sacrificed the mice 60 minutes later to obtain the learning-induced transcripts within the dorsal hippocampus. From those, we looked at which genes were being oscillated and specifically which genes were being upregulated during the day and not upregulated during the night. And we found PER1, which was interesting because we know that's a circadian clock gene that contributes to the circadian rhythm. With that, we then ran qPCR to show that PER1 is being upregulated during the day, but not so much during the night, and showing that same oscillatory pattern that we were seeing with memory. We also took this a step further and wanted to see what would happen if we knocked down PER1 expression during the day when they have that robust memory performance and upregulated it during the night. So when we knocked it down, we were able to induce that memory deficiency, and when we upregulated it during the night, we were able to ameliorate some of those deficiencies. So to conclude, we found that young male mice have an oscillating memory performance across the diurnal cycle, with their best memory being during the day. We found that PER1 is likely contributing to that oscillatory memory pattern. We also found that knocking down PER1 during the day impairs memory, while overexpressing it during the night rescues memory. And now we're looking at specifically the specific PER1 transcript isoforms that may be contributing to memory performance and whether those are separate from PER1's role in the circadian rhythm. And I just want to thank my lab, the Clapis lab, my mentor, Lauren Belfi, and the Astronaut Foundation, and the mice, of course.
Thank you, Megan. Who has a question for Megan?
Yeah, thank you so much for your presentation. That was awesome. I understood a lot about the memory and the mice all working together. As an engineer, I just kind of had a quick question. How do you plan on applying this knowledge that you gained from your research into the future, and would it ever be applied to human medicine?
Yeah, definitely. So I like to think it would be. With some of the conclusions, it could be used as simple as when to study best, like studying. I don't believe that this has been done in humans yet. Obviously, there's a bit more factors, but say this is done in humans and we found that we have better memory performance during the day, then we know to be studying during the day rather than studying at midnight, which I'm sure we all do. And then it can also go on the other side. So with PER1 specifically, if we find that that's the same in humans, we could potentially develop a drug that overexpresses PER1 to rescue memory deficiency in Alzheimer's or dementias. Yeah.
Okay, one more question for Megan.
Great presentation. Thank you very much. Are you aware of any studies in this vein that implant electrodes in the brains of mice and also map that sort of oscillatory patterns that you're seeing in the neurons themselves?
I believe there's another lab at Penn State focusing more on the electric side of things. I wouldn't be able to tell you specifically on that, but if you're interested, you can definitely look at Penn State's research. Sorry, I don't have more specifics for that.
Thank you.
Thank you. And can you meet me later about the oscillating memory performance of husbands?
For sure. I mean, this is
Meet me later. Thank you so much.
We have found differences in females and males.
Oh, there we go. All right, Megan. See you later. Very good. All right, now we're going to have one more presentation before the break, and you've all been fabulous, and we are so on time. So when the break comes, I'll tell you about being on time as well. But in the meantime, let us welcome Mackenzie Jones, and she's going to talk about necrotizing enterocolitis. Woohoo!
Don't need to. There we go.
Good morning, everybody. My name is Mackenzie Jones, and I am a senior biomedical engineering major at Clemson University. This summer, I had the privilege of working at the University of Heidelberg to investigate the role of neutrophil and macrophage interactions in necrotizing enterocolitis. So those are a lot of big words. So to start off with some background, necrotizing enterocolitis is a life-threatening intestinal disease that primarily affects preterm infants. It causes inflammation and necrosis in the gut, and it's believed to result from a combination of things like prematurity, formula feeding, poor gut oxygenation. But the main thing to know is we don't really know what is causing necrotizing enterocolitis. We also call it NEC for short. We think that it might be an immune response because what we're observing in NEC is that the immune system is essentially being triggered, going crazy, and for lack of better terms, the immune system is causing the intestines to eat themselves. So looking closer at the immune system, there are a lot of cells that play key roles in your immune system being able to defend from pathogens. The first cell we want to look at are neutrophils. They are a fast-acting immune cell that respond to infection and inflammation, and we believe they might play a key role in the early pathogenesis and immune response in NEC. The next immune cell we're interested in is the macrophage. These are immune cells that help clear pathogens and regulate inflammation, and they may influence disease severity in NEC. Something important to know about these two immune cells is that they are both phagocytes, meaning they can engulf and digest pathogens, which makes them essential for clearing out infections and initiating the immune response that we see in NEC. So looking closer at the functions of these immune cells, the neutrophils are suspected to contribute to NEC pathogenesis by releasing neutrophil extracellular traps, or NETs. These are a combination of proteins and extracellular DNA that are kind of seen as a double-edged sword. They're essential for the clearance of pathogens in the immune response, but increased levels of NETs have been seen in a lot of very devastating diseases: Alzheimer's, type 1 diabetes, and necrotizing enterocolitis. We've seen it be associated with cell death and tissue necrosis. The macrophages are kind of cool because they can polarize into different phenotypes based on their needed function. And the specific phenotype of the macrophage is actually going to influence how it behaves following its activation. We think that macrophages and neutrophils are going to interact pretty regularly in our immune cell response because macrophages actually secrete DNases that can degrade NETs the way that they need to be degraded for proper immune clearance and to prevent that excessive inflammation. So this brings us to our research question. How do the individual functions and interactions between neutrophils and macrophages contribute to NEC pathogenesis? So to test this, we have developed a three-dimensional organoid model. How we create this is we get small intestine samples from the clinic or the hospital. We take these samples, we wash out cells, and then we suspend these cells in a fancy sort of jello known as a matrigel. We put these in a well plate and grow them up for a week. We co-culture them with neutrophils, macrophages, and then we can stimulate them with something called LPS, which is going to cause inflammation. And what we're trying to do here is essentially see how these intestinal cells are going to behave with the neutrophils and the macrophages. Are we going to see increased levels of proteins, NETs, that sort of thing? So one of the first things that we did was we wanted to observe the different proteins that were released in our organoids, and we wanted to do that through immunofluorescent staining. So here, this is DOPI. This is going to stain for DNA. So this is just showing we have DNA present in our organoid. NE and H3CIT, these are going to stain for proteins that are present when NETs are released. So we see a lot of NETs release in these two images. And then here we just have an overlay so you can see kind of where the DNA is being released, where the proteins are being released, and kind of that NETs are present in our organoid samples. Next, we wanted to quantify NETs formation. We don't just want to see a picture. We want to know, is it actually increased in our organoids that we generate from healthy patients, NEC patients, or is it increased with that inflammatory stimulant that we add? So we performed something called an ELISA. An ELISA is going to detect and quantify specific proteins, antibodies, and other things that we want to look for, and it's going to quantify these through a color change reaction. So our results show an increase in NETs formation in our organoids that we generated from patients with necrotizing enterocolitis versus our donors that were healthy. So you can see our necrotizing enterocolitis donors are the red bar, the healthy are the white bar. We see an increase in that NETs presence, and we also see an increase in NETs presence after adding LPS stimulation. So this is a very brief part of the results that we've gathered from this study, so if you want to hear more about that, please come find me. But some brief conclusions that we can draw is that utilizing a three-dimensional organoid model may provide an accurate in vitro model of NEC, and NET formation can be stimulated by co-culturing organoids with neutrophils, macrophages, and LPS. NET formation seems to be donor dependent. However, organoids generated from NEC intestine samples do show increased NETs formation compared to healthy intestine samples. We're interested in exactly why that may be, but it was also interesting to see that LPS does cause increased levels of NET formation. So that was kind of a proof of concept, making sure that we can stimulate inflammation and then compare that stimulated inflammation to our NEC donor organoids. So lastly, I would like to thank all of my mentors, the DAAD RISE program for funding this research, and I would like to thank the ASF for giving me the opportunity to be here and present this research to you, everybody here.
Well, thank you so much. We have time for one question from Mackenzie.
So great presentation. It seemed like your organoids are relatively blobular. I was curious if you had seen any sort of crypt development in your intestinal organoids and if you're seeing differences within NET action with crypts.
Yeah, so we do isolate the organoids, the epithelial cells from the crypts, so we don't grow our organoids long enough to be able to observe the crypt formation. We typically grow them for maybe a week or two at most before we perform experiments. So I think to observe that, we'd probably have to grow them up a little bit longer, but that could be an interesting study in the future to observe how they interact together.
Thank you.
Thank you so much, Mackenzie. All right, we are so on time, so you get a break because there was coffee. So here's the deal. You have 5 minutes. All right, let's get started with half number 2, and we're going to welcome Adriana McCarty, and she's going to talk to us about something pretty important, preventing respiratory infection. So welcome, Adriana.
Alrighty, hi, my name is Ariana McCarty, and I've been doing work at the University of Colorado Anschutz Medical Campus, exploring the role of Prevotella, which is a bacteria, in preventing respiratory infection. So I'd like to start by talking about pneumonia. Pneumonia still represents an incredible disease burden on our world. It's the 4th leading cause of death, and it's the 1st leading cause of death in children under the ages of 5 worldwide. Streptococcus pneumonia is a specific type of bacteria that is the leading cause of community-acquired pneumonia. And unfortunately, due to serotypic variants as well as antibiotic resistance, our current therapeutic treatments just do not comprehensively address pneumonia as a disease. And that's where the respiratory microbiome comes in. So as so wonderfully explained to us earlier, we are all home to gut microbiomes, just as lemurs are. We are also home to respiratory microbiomes. Our lungs are a home of trillions of flourishing bacteria that can all work comprehensively to do a bunch of different things. The respiratory microbiome does specifically differ between healthy and diseased individuals, kind of cueing to us that it's doing something. There's some role that the respiratory microbiome plays in our lives. Prevotella is a specific type of bacteria that are highly prevalent in the healthy respiratory microbiome, which kind of cued our lab in to investigate maybe how it's playing a protective role against respiratory infection and disease. So I wanted to explore how it's playing a protective role against Streptococcus pneumonia, and I did this in a few ways, some of which I'll share today. First is through a technique called an adherence assay, where this is an in vitro test where I took cells from the lungs and the pharynx, so you get an upper and lower respiratory tract picture, expose them to Prevotella, and then expose them to Streptococcus pneumonia, and essentially just see what grows. From this, I saw that as I added more Prevotella, and specifically Prevotella melanogenica, I saw decreased adherence of pathogens to your respiratory lining. So that very first crucial step to infection, the pathogen is less able to complete. So that's a really exciting result, and we wanted to follow it up with in vivo studies. So we tested in BL6 mice, essentially a similar concept, exposed them to Prevotella as well as Streptococcus pneumonia, allowed that infection to take place over the course of a day, and then took lung tissue, bronchoalveolar lavage samples, as well as spleen tissue samples, and tested them for bacterial burden, and saw once again, really excitingly, Prevotella reduces the amount of Streptococcus pneumonia that were present in those mice after 24 hours, both in antibiotic and germ-free mice. And really excitingly, this coincides with neutrophil populations. We see that Prevotella coincide with a significantly increased population of neutrophils, both percentage and just total count, which are sort of this key immune cell that helps to fight infection. So overall, Prevotella melanogenica is protective against Streptococcus pneumonia infection, which is really exciting. We found at least 2 ways that it does it, through reducing the ability of a pathogen to actually adhere to your respiratory tract lining, as well as increasing the amount of neutrophils, and also through some other data not pictured here, activating those neutrophils more effectively. In the future, we want to investigate interactions between those 2 mechanisms, as well as maybe explore some others, and really explore the potential of Prevotella as a respiratory therapeutic. Specifically, we'd like to bolster data in preparation for a patent application in the coming months. I'd like to go ahead and thank and acknowledge my mentor, Dr. Sarah Clark, for all of her undying support, as well as give a huge thanks to the Astronaut Scholarship Foundation for the opportunity to speak here today and connect with my fellow scholars. Thank you.
Thank you, Ariana. Who has a question?
Sorry, do you mind repeating it in the microphone so our virtual listeners can hear? Thank you so much.
So hypothetically, if someone were to take a heavy course of antibiotics and that bacteria would deplete, they'd actually be at more risk of a lung infection then?
Yes, so that could hypothetically mean, especially if those antibiotics did target Prevotella. One exciting thing that we found, though, is that these trends are demonstrated in heat-killed Prevotella, which is really crucial in terms of therapeutic development if you're not introducing a live organism to someone else, but that could potentially have implications where antibiotics might not be reducing that activity as much.
And one more question.
Give a comment from our virtual audience. Someone really loves all the gut microbiome research going on, so that's really exciting.
One more question. Anybody? All right. Well, thank you so much, Ariana. Next, we would like to welcome Harini Ram, and they are going to talk to us about high-grade serous carcinoma. Welcome, Harini.
Hi, everyone. My name is Harini, and I'm a rising junior at the University of Michigan studying molecular, cellular, and developmental biology. And today, I'm going to be talking about how miR-181A and MYC drive carcinogenesis and metastasis in high-grade serous carcinoma through enhanced purine metabolism. And so to start off with a little bit of background, I study high-grade serous carcinoma, which is the most common and lethal form of ovarian cancer. Unfortunately, over 75% of patients are diagnosed when the disease has already spread throughout the abdominal cavity, what we call metastatic spread. And most patients respond to chemotherapy regimens initially, but 70 to 90% relapse and develop recurrent disease. Because of this, scientists are working to discover the early events of progression that contribute to this aggressive nature. And so far, we've identified a few key hallmark events, including mutation of the tumor suppressor gene P53 and formation of these precursor lesions, what we call stick lesions. However, the molecular mechanisms that actually turn these fallopian tube cells into these aggressive late-stage metastatic cancer cells remain largely unknown. And so to investigate this, my lab previously identified a molecule, miR-181A, to play an important role in ovarian cancer progression. So when we all think of RNA, we think of messenger RNA, which serves as a guide to encode for proteins that carry out all the functions in our bodies. But microRNAs are different. MicroRNAs are small, regular regulatory RNAs that influence gene expression for multiple genes at once. And they do this by binding to the ends of messenger RNAs and causing them to be degraded or blocking their translation into proteins. This allows them to influence a wide variety of biological processes, including those involved in cancer, such as uncontrolled cell division and evasion of cell death. In addition to miR-181A, I also study the well-known oncogene MYC. MYC is a transcription factor, meaning it's a protein that binds to DNA and turns on genes involved in cell growth and division. Interestingly, we see here that MYC is most amplified in ovarian cancer when compared to all other types of cancers. And so from patient tumor databases, we see that 11% of high-grade serous carcinoma patients have high MYC and miR-181A, and that these 11% of patients have significantly worse disease-free survival. What that means is that these patients will relapse and develop that recurrent disease a lot faster. And so to move beyond looking at patterns in patient data, we needed a way to model this in the lab. We engineered 4 different cell lines to model this, and we started with these non-cancerous fallopian tube cell lines, and then we created 4 different versions: a control version, a version with 181A overexpression, a version with MYC overexpression, and then a version with the combination overexpressed. And then when we did a phenotypic assay called sphere formation assay, we saw that the combo cells had an increased sphere formation rate. And then when we implanted these cells into mice, we see that the cells with both the 181A and MYC formed tumors at a much faster rate with increased volume as well. And so this brings us to my main question. What are the downstream pathways of miR-181A and MYC overexpression that drive these aggressive phenotypes? And so to identify these clinically relevant drivers of this aggressive phenotype, I did something called GSEA, stands for Gene Set Enrichment Analysis. Instead of looking at changes in individual gene expression across conditions, I'm trying to see if we can look at entire groups of related genes and see if certain groups are collectively more active in one condition over the other, specifically the combo cells. And so I applied the GSEA to both patient tumors with high MYC and 181A and our cell line with high MYC and 181A and overlapped them, and found that purine metabolism was significantly enriched. And so what exactly are purines? Purines are these compounds that serve as the building blocks of DNA and RNA, are involved in protein synthesis and cell signaling, and ATP serves as the primary energy currency in the cell. And so within purine metabolism, we found that IMPDH2 is a critical metabolic vulnerability. It catalyzes a step from IMP to XMP, which forms the nucleotide GMP. And so we see here that when we treat ourselves with MPA, a drug that inhibits IMPDH, the combo cells are significantly more susceptible to that decrease in viability. We can see that there's a sharp drop in their growth in the combo cells compared to the other 3 groups. And then when we implant these combo cells with lower IMPDH2 into mice, they form tumors at a much slower rate and with a decreased volume. And so for future directions, we see that MPA is very effective at inhibiting cell growth and slowing their division. However, this is called a cytostatic effect. And in order for a therapy to be truly effective, we want it to be actively killing the tumor cells, what we call cytotoxicity. So currently, we are doing a high-throughput drug screen, treating our cells with MPA in conjunction with thousands of different FDA-approved compounds to find compounds that synergize with MPA to induce that tumor cell death. And so with the future of cancer progressing towards personalized medicine, my goal is to contribute to the discovery and implementation of metabolic therapies that target purine metabolism for aggressive high-grade serous carcinoma. And I would just like to give thanks to my lab, the DeFeo lab, as well as our collaborators, funding from the Rogel Cancer Center, and ASF for all your support and guidance.
Thank you so much, Harini. Who has one question for Harini? There's one in the back. I see that hand up.
Thank you for your research. I'm curious about polycystic ovarian syndrome and HPV, and if there's any correlation with this research and the development of cure for those or treatment.
So I think polycystic ovarian syndrome is primarily like although it involves cysts, I don't believe that these are like malignant and can progress to these like lesions that we see in precursor high-grade serous carcinoma. And for HPV, actually, I think there is a correlation that's still being discovered because HPV originates from a virus, and we haven't really, research in high-grade serous carcinoma hasn't really shown that viruses can contribute to the progression, but I think it's like an emerging field.
Thank you so much. Next, we would like to welcome Alara Berkman, and she is going to speak with us about Ewing sarcoma cancer.
It's a lot scarier up here than people are letting on. Hi, everyone. Nice to meet you. My name is Alara Berkman, and I'm an undergraduate researcher studying biomedical engineering at the University of Southern California. I'm a fellow in the Amatruda Lab at Children's Hospital Los Angeles, and I work for the California Institute of Regenerative Medicine. Today, I'd like to discuss my project on the characterization of Ewing sarcoma metastasis in zebrafish models. Okay, great. So Ewing sarcoma is the second most common pediatric bone tumor affecting children primarily from ages 5 to 15 and localized in the bone, bone marrow, and lungs. There are around 250 cases in the US alone each year, and it is caused by a chromosomal translocation between the EWS gene and the FLY1 protein DNA binding domain, which causes a very aggressive fusion oncogene implicated in DNA replication and repair. While the 5-year survival rates for EWS have improved significantly, they kind of float around 70% pre-metastasis. Following metastasis, they drop to less than 25% when involved in bone marrow and lung tissues. So the purpose of my project is, due to a lack of reliable in vivo models, we haven't been able to understand how or why EWS metastasizes, which has significantly limited our ability to develop a targeted intervention for these children. I aim to establish a xenograft zebrafish model to elucidate cell intrinsic and tumor microenvironment factors that may be contributing to EWS's metastatic behavior. I'll be specifically looking at 2 cell lines, CTLA-9 and CTLA-10, and comparing them based on proliferation due to its role as a metastatic hallmark of cancer progression. I compared CTLA-9 and CTLA-10 in particular because a very important fact about these cells is that they were biopsied from the same patient. So CTLA-9 is from the primary tumor or the site of origin, and CTLA-10 is from a metastatic tumor that was taken later on. The reason that my lab uses zebrafish, there's a number of reasons, but primarily they have very high fecundity or birth rates. They develop completely transparent for the first 14 days of their growth, which makes them very easy to image. And for me in particular, since I'm looking at metastasis, I need to be able to understand where is the primary site of origin, where did this tumor start from, because I need to be able to compare primary and secondary sites. So when I xenograft, I'm able to deduce, well, obviously I injected here, so I know where those cells started, and I can look at distal tissues based on that location. Additionally, they are an effective representation of human cells because I will be using human, not mutational zebrafish cells. And we've seen in multiple labs that have demonstrated that drug work on human cells in zebrafish has shown very high efficacy and response to drug treatments compared to how it would actually behave in a human. For my procedure, I started by injecting in the perivitellin space, which is just known in our field as the most robust site on the zebrafish, and then I incubated them for 3 days at 34 degrees Celsius. The reason I use 34 degrees is because zebrafish body temperature is 28 degrees, humans prefer 37 degrees, so the fish or the cells died at 33 and 35, so we went with 34. Next, I screened the fish based on survival engraftment, which just means the human cells were able to survive over the incubation period, and whether or not they were able to metastasize to distal tissues. So you can see in the fish down below, it has a very distinct PBS tumor, and it has a lot of involvement in the tail. Then I used immunohistochemistry staining, looking specifically at filamentous actin GFP, which just means the cytoskeleton of the human EWS cells. And I also stained for phosphohistone H3, which is very specifically expressed during mitosis. Lastly, I used Imaris to create 3D models that could demonstrate the volume of my tumors and the number of actively proliferating cells. And what we were looking for is an overlap in those cells. So you can kind of see there's these red cells within the tumor mass, and that demonstrates that that is an actively proliferating human EWS cell, which means that not only have these cells managed to engraft in the fish, they have the potential to metastasize further and grow within the fish, creating more aggressive tumors. So for the quantification in the yolk, normally I took the percentage of cells out of the human EWS mass, but the staining was too muddled, so the yolk volume was the quantification that I used. For my results, I found that in survival and engraftment rates, there was no statistically significant difference between the primary and secondary tumors. However, in metastasis, I did see that there was no overlap in the error bars. So using a Welch's t-test of unequal variance, I confirmed that there was a statistically significant difference in the metastatic occurrence between CHLA9 and CHLA10, which I will discuss in a moment. In our immunohistochemistry staining, we saw just very obvious distinctions between the behaviors of the cell. CHLA9 has a very tight tumor, whereas CHLA10 is a little more broad. The cells are a little more distinct. You can see their boundaries better. And there's also very different characteristics in the tail. So CHLA10, as I said, it has a lot more volume involved, and it demonstrates a lot more metastatic growth. So it was able to develop a stronger tumor microenvironment in the caudal hematopoietic tissue of the tail, which is indicated on the slide. And in summary, we've kind of seen that these CHLA10 cells, these secondary tumor cells, have developed some kind of post-metastatic survival adaptation that is enabling them to react better in foreign tissue. So the distinct environmental preferences are indicating that maybe there's some kind of mutation that they've developed that we need to examine further by either RNA sequencing or looking more into the mechanisms that these cells develop. Additionally, for my analysis of the proliferation in the tail, neither of them showed significant differences in the occurrence of that double positive EWS proliferating cells in the tail. However, CHLA9 had absolutely no proliferation in the tail, and CHLA10 had a little bit. So that's hope for the future. The future directions of my project right now, I want to do a time course analysis on both CHLA9 and CHLA10 to see maybe CHLA9 is developing proliferative tissue in the tail, but we just haven't seen it because it's too late within the process. Additionally, I'll be using photoconversion to see once they get to the ventral side of the fish, can they move dorsally and metastasize further from that tail site? Which would indicate that they are able to develop further metastases throughout the body, and that may help determine any possible secondary metastatic events throughout humans. These are my references. And I would just like to thank the Astronaut Scholar Foundation for being so welcoming. I'd like to thank Carol for really supporting me throughout this experience. And I want to thank my lab and the Compass program that I'm a part of back at USC. Thank you.
Thank you so much, Alara. We're going to skip questions to stay on time, but you know what we have to do? It was Alara's birthday yesterday, and guess what? She turned 21. Woohoo! Happy birthday to you. Happy birthday to you. Happy birthday, dear Alara. Happy birthday to you. Yes.
Thank you.
And if you thought it was scary up there, you didn't know I had the microphone. All right. Thank you so much. Now let's welcome Caroline Nguyen, and she's going to talk to us about more advanced cancer immunotherapy. Hello, Caroline. Thank you.
Hi everyone, my name is Carolyn Nguyen. I am a rising senior at the University of California, Berkeley, and I work in the lab of Dr. James Nunez in the Department of Molecular and Cell Biology. And we study epigenetic editing with CRISPR in order to advance cancer immunotherapy. So CRISPR-Cas9 technologies are a powerful gene editing technology that allows us to create precise modifications anywhere within our human genome. And the advent of CRISPR-based genome editing technologies have been really transformative for biomedical research and therapeutic development. However, a caveat with Cas9 therapies is that recent studies have found that when the cell tries to repair the double-stranded DNA break that CRISPR-Cas9 creates, it can create some mistakes like large deletions, inversions, translocations, and even aneuploidy, which eventually can cause cancer. And so our lab is focused on the epigenome, or the modifications kind of on the outside of the DNA that change its accessibility to different transcription machinery. And so we've built this tool called CRISPR-Off that uses a catalytically dead version of Cas9, so it can still guide to where you want to go in the sequence. But instead of cutting the DNA, we fuse a DNA methyltransferase and a histone modifier to our dCas9. So that can add methylation marks that'll close up the DNA, and so that way we're kind of bypassing this double-stranded DNA break and instead closing off that DNA to machinery that can transcribe and translate it. And so in practice, what this looks like is if we add our CRISPR-Off epigenetic editor into cells and monitor gene silencing, we can see that even though the CRISPR-Off expression is transient, meaning it leaves the cell within a couple of days, that memory of methylation is maintained over a course of time. And so this is a one-shot delivery with sustained silencing, which is really exciting for therapeutic use. And so my goal is to see if we can translate this into more primary cell lines, since that last one was an immortalized cell line. So we took here primary T cells that were isolated from patient samples, added our epigenetic editor in the form of mRNA, and monitored gene silencing. And this is data as of day 30. So this is a flow cytometry plot. So each dot means it's one cell. And we drew this gate based off of our untranslated control sample, which means that we didn't treat it with any mRNA yet. And so all of the dots outside of the gate means that the gene is not silenced. But however, when we added our mRNA, we could see most of these cells shifting into the off gate, meaning that that gene has been successfully silenced within that cell. We're also hoping to eventually go after genes that are upregulated in T cells that cause exhaustion. So right now we're just targeting a non-essential gene just to show that this works, but eventually we want to go after some genes that are upregulated in exhausted T cells. We also have tried this in stem cells, since stem cell therapies are a good way to get a renewable cell source for therapies for patients. And so when we added our epigenetic editor mRNA to our stem cells, we could see as of day 2, it looks like the cells are starting to shift into that off gate, but as of day 21, they're kind of back into the on state. And so this is a problem that we're facing right now, is that these CRISPR-Off stem cells seem to reactivate over time. However, we also have tried to redose these cells, and we've seen actually that redosing can lead to stronger gene silencing. So most of these cells are shifting into the off gate as of day 2, and as of day 10, most of them are still kind of in the off gate. So that was promising for us. And eventually, we want to see if this has potential to advance cancer immunotherapy using these epigenetically modified iPSCs, or induced pluripotent stem cells. We'll eventually differentiate them into different cytotoxic T cells, and that will hopefully go on to fight cancer cells better. But yeah, just wanted to acknowledge my lab. It's been an incredible time in doing research with the lab, and also thanking the Astronaut Scholars Foundation for this experience. I'm very grateful to be here. But yes, thank you.
Thank you, Caroline. Who has a question?
Thank you, Caroline. A question about other applications for other epigenetic diseases, such as sickle cell.
Mm-hmm.
You see this as a possibility, or is that in a completely different realm?
Yeah, I think for right now we're focusing mostly on what we can target with the T cells and boosting like the cytotoxicity of T cells. Right now, I think for CRISPR, there's a good possibility for therapies within like lymphomas and blood cancers. We're not trying to go towards like solid tumors right now just because it's a little bit more difficult to kind of engineer the T cells that way. But yeah, that's definitely a possibility that we're thinking of in the future.
One more question.
I have a quick question. Do you see the same efficacy for histone acetylation with your method?
Yeah, so yeah, right now we're focused on methylation, but an arm of our lab is actually also looking at activation. So for diseases where there is an underexpression of genes, we're hoping to kind of send in our tool to add those acetylation marks. But yeah, that's kind of an area that our lab has not yet explored. But yeah.
Thank you so much. Next, we would like to welcome Kritik Tella, and we're going to talk about Richter's transformation. Could be another band to compete with the other one. Welcome.
All good? That was smooth. All right. Hello, everyone. My name is Kritik Tella. I'm a rising senior at The Ohio State University. I've been working for the past 3 years in the La Palambella Experimental Hematology Lab. Here we use genetic and translational approaches to better understand and treat blood cancers. My specific project that I'll be talking about today revolves around an aggressive lymphoma called Richter's transformation and figuring out any therapeutic vulnerabilities that we can target. But first, a little background, or before getting into Richter's, we first have to talk about its precursor disease, which is chronic lymphocytic leukemia, or CLL. CLL is a cancer that starts in the B cells, which is a type of immune cell, in white blood cell in your immune system. These cells grow and build in places like the blood, bone marrow, and lymph nodes, and they don't work properly to fight infections. It's actually also the most common leukemia in the Western world, but thankfully, due to recent advances in targeted therapies, patients have had greatly improved outcomes. But however, CLL is still not incurable. And in about 2 to 10% of these cases, the CLL can actually change or transform into this more aggressive cancer called Richter's. And once this happens, it becomes very difficult to treat, and survival is often measured in months rather than years. The main problem with Richter's is that we don't fully understand its unique biology because it's sort of a transformation from another cancer, which has made it really hard to find any sort of like target that we had found for CLL, for example. Our solution to this was kind of piggybacking off the last talk, running a genome-wide CRISPR knockout screen. Despite the really complicated procedure on the slide, in its essence, CRISPR screens essentially turn off each gene one by one in our cells, and we basically were able to infect Richter's cell line and a CLL cell line with this viral library of single guide RNAs that can target over 19,000 genes. And after infecting these cells, we were able to track which gene knockouts caused cell death by sequencing these guide RNAs over time. And the way it works is cells with non-essential gene knockouts will survive and grow, while cells with missing essential genes will ultimately die off. So by comparing these abundances, we can identify which genes are critical for survival. But you might be thinking, what about the actual genes that we need for everyday survival for healthy cells? And in order to combat this, we can filter out these core essential genes by comparing it to a known database. This will leave us with a set of genes that are uniquely enriched in Richter's that can ultimately pave the way for future treatments. And based on this whole procedure, we then performed this screen in both, as I said, Richter's and CLL cell lines. And there's also shown above, we found there's a huge set of genes, and then using some software, we were able to map it to the pathways that they belong to. And the reason that I boxed oxidative phosphorylation is that this was kind of our aha moment. So in the rest of the pathways shown, like nucleotide excision repairs, these showed up in our CLL screens. So what was unique to Richter's was this pathway of oxidative phosphorylation. And the ultimate goal that we were trying to aim for here was to find something that was unique to Richter's. So this was exactly it. And a little background on oxidative phosphorylation is, like many of you might have learned about in your intro to biology classes, it happens in the mitochondria, which is the powerhouse of the cell, of course. And it's basically a process that generates ATP, which is the source of energy in our cells. And logically, we thought, what would happen if we block off this pathway with some cool clinically relevant inhibitors, namely ICS and IM, which are already in clinical trials. And some preliminary results are shown above. So we tested out these inhibitors, and we found that ICS had the greatest therapeutic benefit based on the cell viability assay, as you can see in the bottom black bars. So this was our drug of choice moving forward. Furthermore, what was really cool and sort of assuring, in another cell viability assay, we performed this in both our Richter cell line, which is URT1, and as well as our CLL cell line, HG3. And we saw that our drug ICS was actually selective in our Richter cell line, and it was also dose dependent. We can see in the 10 nanomolar and the 100 nanomolar bars that it affected URT1, our Richter cell line, a lot more significantly compared to our HG3 CLL cell lines. So this is also another sort of evidence that this is a very promising RT-specific therapeutic strategy. So ultimately, this was our first, as I mentioned, frontline therapy that could be used in the future. Another sort of impact that it creates is in terms of personalized cancer care, it can be used as a metabolic signature that can identify CLL patients most at risk for transformation. Some future work that we want to do moving forward is validate OXPHOS inhibition in more living system models like mouse models, as well as test drug combination therapies to minimize toxicity and maximize effectiveness. Huge shout out to my mentors in the La Palambella Experimental Hematology Lab, and Dr. Ethan Whipp, my grad student mentor, and of course, huge shout out to ASF for creating such a fun and inspiring weekend for us. And thank you all for listening.
Thank you so much. Thank you, thank you. Who has a question? I see one right up front.
Thank you so much for that presentation. That was awesome. So I'm curious about, it's interesting that you're inhibiting OXPHOS just because I feel like normally for cancer, they do a lot of glycolysis instead. So do you think that inhibiting OXPHOS could have any negative effects like for that certain type of cancer just because some cancer cells rewire in order to really depend on glycolysis instead?
Yeah, that's a great point. So actually, there's a lot more data that I was not able to show here, but Richter's, like you might assume, there is a glycolytic shift, as with most cancers. But the thing is with Richter's is that it's heavily reliant on OXPHOS compared to other cancers, like for example CLL. CLL is very big on fatty acid oxidation as using getting more energy, but actually Richter's is, instead of doing that, since it's a really aggressive cancer, it has an even higher energy input that's required. So that's why we think it's more reliant on OXPHOS. But yes, there still is a glycolytic shift that we can maybe target in the future as well.
Good. One more question.
I was just curious about why you think that the specific drugs that you were mentioning, like the IACS or whatever that was, like, why do you think that it is specifically more selective than the other ones that you tested?
That's a great question, and a great field that I really don't know much about. These are all made by big pharma companies, so I mean, I honestly don't have a good answer for you.
All right, well, thank you so much, Critic. Next, we would like to welcome Caleb van Watermelunen. I hope I said that right. Got the Caleb part right. So we're going to talk about deep cerebellar nuclei. Woohoo!
Woo! Wait. Oh, this is so cute. Hi, good morning, everybody. I'm Caleb van I'm Caleb van Watermelunen, and I'm from the University of Minnesota, Twin Cities, studying neuroscience and biochemistry. And what I've been doing is looking at differentially expressed genes in the deep cerebellar nuclei of spinocerebellar ataxia type 1 mouse models. To begin, what is spinocerebellar ataxia type 1? So spinocerebellar ataxia type 1, or just SCA1, is this progressive neurodegenerative disease that has this gene called ataxin-1 that is central to its pathology. So in people who do not have SCA1, this ataxin-1 gene has a non-pathological number of these CAG repeats within it, where these CAG repeats are these trinucleotide sequences within that ataxin-1 gene again, that are eventually translated into glutamine residues. And the idea of having too many of these glutamine residues within that ataxin-1 protein is that the final translated protein, or gene, excuse me, is going to be too sticky and contribute to like cell toxicity, which we don't like that, that's not fun. So again, in people who don't have SCA1, there's going to be a normal ataxin-1 protein, you're going to have normal Purkinje cell survival, a happy, goofy cerebellum, super cute, and then you're also going to have a healthy phenotype. In people who do have SCA1, though, there is a pathological number of these CAG repeats, specifically more than 39 within that ataxin-1 gene. This is going to translate into a sticky ataxin-1 protein, ultimately killing these Purkinje cells, not through explosions, I promise, degrading the cerebellum, and then producing an SCA1 phenotype. This SCA1 phenotype is going to be characterized by things such as nystagmus, lack of coordination, and even cognitive impairment. So why am I looking at the deep cerebellar nuclei instead of like Purkinje cells, other brain regions, like other cell types? So the reasons I'm looking at the DCN, or the deep cerebellar nuclei, are going to be first that there's a lack of DCN research within these SCA1 mouse models because it's all been on Purkinje cells. Second is going to be the identifying possible molecular mechanisms of SCA1 disease onset. Third is going to be, there we go, understanding or discovering pharmacological targets to treat SCA1. And finally, looking at the behavioral and phenotypic effects of pharmacological alterations. For my actual project, a quick preface is going to be the mouse model I'm working with. So the mouse model that I've been using is called an ataxin-1 154Q mouse, which just means that these mice have 154 of those CAG repeats within that ataxin-1 gene, which again is significantly higher than that 39 CAG repeat threshold for the SCA1 disease onset. The reason for using these 154Q mice is that they exhibit SCA1 symptoms that you would see in humans, primarily severe ataxia and early onset of death. And it also makes it easy to look at changes in gene expression as well as phenotypic changes that come with pharmacological manipulations. As for my actual project, there are 4 main steps. So the first step of my project was looking at previous RNA sequencing data, which measures relative mRNA trends, or as some people say, mRNA, and identifying candidate differentially expressed genes, or DEGs. So and this was between the 154Q and wild-type mice, with an emphasis on pharmacologically relevant DEGs, or those that could be targeted by ligands or drugs. I've identified over 120 DEGs, focusing on 2 called CRHR1 and IL-20RB, which are genes that encode for receptors related to stress and neuroinflammation, respectively, and both were downregulated within these 154Q mice. So based on this cool little RNA-seq data, I thought it'd be fun to do some pharmacological tests to see if I could administer these ligands and rescue the SCA1 phenotype, or even like reproduce that. But before I could actually get to that step, because my PI said I was going too fast, I had to confirm the RNA sequencing trends. To do so, I took DCN punches, so literally punched out the DCN, or the deep cerebellar nuclei. I then isolated the RNA, turned that RNA into DNA, and then ran qPCR, and I did confirm that both genes, or relative mRNA level, was downregulated in these 154Q mice. I'm currently working on protein expression trends in 2 ways. The first way I'm looking at protein expression trends is I'm taking, again, these DCN punches, creating protein lysates, and then doing Western blots, as well as taking whole brain coronal slices of the deep cerebellar nuclei, or DCN, and then doing immunohistochemistry to stain for my receptors of interest, and then doing immunofluorescence imaging. To which, hopefully, if science decides to science this semester, I can actually confirm these protein expressions trends. If the protein expression is significantly altered within these 154Q mice, I'll then move on to my pharmacological tests. So to do the pharmacological test, I'm going to hook up these 154Q mice with osmotic pumps. Super fun. So these osmotic pumps are going to be these controlled drug delivery systems that can administer these ligands to my receptors of interest. After these use of osmotic pumps, I'll then look at changes in phenotype, mRNA expression, protein expression, with tests such as behavioral, for example, rotarod, Western blotting, and then immunofluorescence imaging, and like previously mentioned, qPCR. Overall, my project has been identifying pharmacological targets to treat or ameliorate the effects of SCA1, contributing to DCN research, because again, all research regarding SCA1 has been focusing on like Purkinje cells. And finally, understanding the molecular mechanisms of SCA1 disease onset. Some quick acknowledgements are going to be my bench mentor, Krista Clark, who is super cool and recently became a PhD candidate regarding her work with the DCN and addiction. And then my PI, Dr. Maria Spatanovich, who has blessed us today with Louie's presence, who is this cute little pupper, super cute. And she's honestly the reason I still want to pursue a PhD. I never saw myself in science, and here I am. So these people are amazing, super supportive, and I love the lab. So yeah, thank you guys for listening, and does anybody have any questions?
Wow, thank you, Caleb. Who has one question for Caleb?
Hi.
Oh, come on, after all that, it's gonna be a question.
Oh my gosh.
There we go.
No. Oh, silly.
Excellent job, Caleb.
Hi.
Loved it. Okay, so what is the advantage to those osmotic pumps for delivering the RNA as opposed to just like IP or oral gavage or something?
Or IP or
Sorry, interperitoneal injection.
Interperitoneal. I honestly.
Just like in the tummy.
Oh, you're good. I honestly am using it just because that's what our lab is familiar with. We've been doing we've done previous drug tests with like osmotic pumps and not really like IP or other ways of doing it. So I think for the sake of it, or me doing it, is just we have the protocol, we have the technique, it's easy to do. Nothing like super major about that.
Great, thank you. And by the way, I live in Minnesota, Sunday dinners.
Yeah.
All right. Thanks, Caleb. All right, next we would love to welcome Natalie Callahan, and she's going to talk to us about substrata stiffness. Welcome, Natalie.
All right, hi everybody. My name is Natalie Callahan, and I am an incoming senior at Colorado State University. And the research that I'm talking about today is the role that substrate stiffness plays on fractional killing in cancer cells, with a specific emphasis on cell cycle progression. Okay, cool. So fractional killing is the phenomenon where a consistent fraction of cells survive therapeutic interventions, regardless of increasing concentration or amount of that chemotherapeutic drug. And these surviving cells are very important because they are what allow the cancer to repopulate and metastasize throughout the body. And so when thinking about a solid tumor specifically, most are heterogeneous in terms of their extracellular matrix stiffness, meaning that they are not the same stiffness throughout the tissue, with generally a softer core with increased stiffness towards the periphery. And this has created a clinical treatment challenge, as having that different stiffness can affect the mechanical response of cells to various chemotherapeutic interventions and lead to this fractional killing phenomenon. And so my research specifically investigates how stiffness impacts fractional killing and what mechanism could be behind this. And so for my experiment specifically, I used a PDMS gel with varying stiffnesses in order to mimic different 2D extracellular environments. And then through the use of doxorubicin as the chemotherapeutic agent, I was able to induce apoptosis or cell death. And then confocal fluorescent microscopy was used in order to visualize those cell death dynamics, as well as the internal cellular structures in real time. And so when comparing the activation of caspase-3, which is generally a reliable indicator of cell death, and doing so on a single cell basis, where each line on one of these graphs represents a single cell and its caspase activation over time, we can generally see much less activation on cells that are cultured on that soft gel, compared to those on a very stiff plastic. And then additionally, in a graph that's not shown here, cells that are cultured on an intermediate stiffness gel show activation that is slightly greater than that on the soft gel, but definitely less than that of the plastic. And so overall, this demonstrates that a softer substrate results in less overall cell death, compared to a stiffer substrate, which results in more cell death. However, at this point, we didn't really know why that was. And so when performing a simple cell cycle analysis using flow cytometry, in the two graphs on the left, we can see the cells that are grown naturally on each substrate, and we see relatively equal amounts of each cell in each cell cycle phase. However, when doxorubicin or a chemotherapeutic agent is added, we see a steep decrease in this S cell population as shown by this lower peak here. And so this is just demonstrating the exact point in which our drug is able to actively kill these cells, which is in this S phase. And so this brings up an important point about how cell cycle progression into the S phase is what allows our specific drug to be able to target them. And so most cell types are known to proliferate or replicate faster when they are cultured on a stiff substrate due to upregulated signaling, which allows each cell to reach this S phase point sooner. And so this increased proliferation is actually what we believe to be contributing to more cell death. And so when using a thymidine block to synchronize all of our cells to that S phase boundary, and then adding our chemotherapeutic agent, there is no longer a statistical difference in the caspase-3 activation or cell death across the substrate stiffnesses. And this basically tells us that by taking out cell cycle progression as a factor, fractional killing is really no longer induced by substrate stiffness. And so overall, the main point of this research that I discussed today is that increased stiffness of the substrate or extracellular matrix results in a greater fraction of cell death, and that cell cycle progression is a significant contributing factor to the rate of fractional killing. And therefore, temporarily increasing the proliferation rate of cancer cells before drug application, while in theory harmful, could actually potentially result in increased therapeutic results. And so, in addition to further three-dimensional and tumoroid studies, I have plans to do further exploration into the characteristics of these surviving cells, as well as with collaborators using machine learning algorithms to determine if initial cell shape could potentially be indicative of cell fate. And so I would quickly just like to acknowledge my entire lab, especially my PI, Dr. Soham Ghosh, as well as give a big thank you to ASF for giving me the opportunity to speak here today and connect with so many of my peers. So thank you.
Thank you. Thank you, Natalie. Who has a question for Natalie? Oh, there's got to be a question. Kelly, Blake, ask a question.
Hold up for a microphone for me. That way the virtual people can hear you. Thank you. Appreciate it.
Curiosity, when you're moving forward with the study, are you focusing on a particular type of cell, or is it any and all?
At this point, we're just using a general HeLa cell model.
Okay.
So just pretty generally looking at the mechanistic differences of the stiffness.
How do you see applying this research into a therapeutic model?
Yeah, so obviously focusing more on specific cell types or cancer cell lines. Specifically, we're looking at potentially melanoma or different liver cancers. And so tumoroid models, and then in theory, in vivo mouse studies.
Yes.
Thank you, Kelly, for letting me pick on you in the best way possible. We have one other question in the front row. The front row is pretty empty. Next session better be filled.
Do you have any idea what types of machine learning algorithms you want to use for the future research? Or?
Yeah, the idea is to use a random forest model to look at different, basically cell phenotype through imaging purposes. Yeah. Wonderful. Thank you.
Well, thank you so much. Next, we would love to welcome Jane Santamore, and she's going to talk to us about hemodynamic loads. Welcome, Jane, and welcome back from last year.
Good morning, everyone. My name is Jane Santamore. I am a senior at the United States Air Force Academy. This past summer, I worked as an undergraduate research intern at Harvard University's Disease Biophysics group, and today I'm going to be talking with you all about controlling hemodynamic loads in tissue-engineered cardiac ventricle models, which sounds like a mouthful, but I will break it down. It will be easy to understand. Okay, so first we start by talking about the heart and its structure. So there's two parts of the heart that I'm going to be focusing on. The first are your ventricles, pretty straightforward. They're responsible for pumping blood throughout the body. Second off are your valves. They open and close, and they ensure that blood is flowing in a singular direction throughout the circulatory system. So now that we've talked about the structure of the heart, right, we can move on to quantifying its functionality, how well it's doing essentially. We can use something called a pressure volume loop, which is here on the right. And essentially you don't need to know like what goes into it or how it works. All you need to know is that valves opening and closing creates the shape of a pressure volume loop. So basically we can't have a pressure volume loop without valves. The other thing that's important to address too is that your heart is constantly changing, right? For example, if you're an athlete, if you're getting stronger, if you're lifting weights, your heart is going to be adapting to that, which is why it's important that we discuss hemodynamic loads, right? When you put your heart under additional stress, additional loads, in this case, I have them termed as, as preload and afterload, which are highlighted at the top here, that is going to cause changes in pressure volume loops. So essentially, a big takeaway from this slide, your heart adapts, leading to changes in pressure volume loops. So now we need to talk about how we're going to study this, right? The lab that I worked at over the summer, they create their own models of ventricles by having cells, culturing them, seeding them onto a scaffold. And as you can see at the top here, they contract, they beat. The only issue in the context of studying pressure volume loops is they're all open at the top, right? They're missing all of those valves across the top, so you cannot obtain a physiological pressure volume loop with the current models. So that's where something called a bioreactor comes in, which is here at the bottom left. And essentially what that does is it incorporates valves. It's like a little plastic structure. You place the ventricle in it, and it incorporates artificial valves in order to establish those pressure volume loop capabilities. So for my specific project over the summer, we asked whether preload and afterload, those stressors on the heart, would be sufficient to induce a stronger tissue-engineered ventricle, right? So when we were growing those ventricles in the lab, can we make them stronger by culturing them under those stressful conditions? And we broke it down into two steps. First, you need to be able to synthesize those ventricles. And the second step is to take those ventricles and put them into a bioreactor. So here's just some goals. I'll skim over those. And then how do we even begin making these ventricles, right? You start with a 3D printed CAD mold. You'll fill it in with some PDMS, which is like this flexible material, and you'll place sugar on the inside. And then you're left with quite literally a sugar model of the left ventricle at the bottom there. And then this is the fun part. We use basically what's like a cotton candy machine. It has all of these fibers coming out, and it's spinning super duper fast, and it's depositing those fibers onto that sugar mold that I mentioned earlier. So once you're finished with that, you get something that looks like this. You can see the fibers on top of the sugar there. There's some post-processing that's done, and eventually you're just left with a very flexible fiber scaffold. So once we have one of those, we need to put cells on it, right, so that it becomes living. In this case, we use two different types of cells. The first one are neonatal rat ventricular myocytes, basically rat heart cells. And as you can see, I have a video of one beating on the right here. And then we can also use human cells, or iPSCs, induced pluripotent stem cells. And I have another video of one of those beating on the right here. It's a little bit weaker, but you can still see it beating. And then how do we quantify, right? So once you're making those ventricles, how do we say, did it work or not? Basically, you can look at it with particle image velocimetry, see how strongly it's beating. Basically, you're looking at how fast these particles are moving. And then we also, circling back to the pressure volume loops before we dive into the bioreactor, we just wanted to make sure that you can get some sort of pressure volume loop from the ventricles that we made. There's a lot of noise going on. The system is kind of complicated, but you can get that kind of pressure volume loop structure right there. So now we dive into the actual kind of materials portion of this, or building the bioreactor. There was a past bioreactor done, but it was for an old ventricle that was open at the top. So now, in accordance with our updated ventricle that's closed off at the top and has the ability to incorporate valves into it, we needed a new bioreactor, which led to the design we built on the right. It has an interventricular loop and an out-of-ventricular loop to circulate nourishment for the cells and kind of establish those conditions for favorability for the cells to grow in. And then we set it up for culture, right? So we're growing those ventricles that we made inside of the bioreactor. We're using a pump to kind of circulate fluids throughout it to sustain life in those ventricles. And for this case, we set it up under continuous perfusion, meaning it's not pulsatile. It's not really beating like a heart yet, but it's continuously circulating. And then we quantified the functionality, again, with particle image velocimetry. At the top, we have our one grown in the bioreactor, and on the bottom, we have just a control ventricle that was cultured in just a regular dish, so the media or the liquid is not flowing, it's just sitting. In the future, we ran out of time. We did this over the summer. It was a really short time to try and culture a lot of cells, but in the future, we would like to move on to culturing under pulsatile conditions to even more closely replicate how your heart works, essentially. And in summary, here's just some skills, things I learned from over the summer. And I would like to wrap it up with a very huge thank you to everyone from the Disease Biophysics group, especially our PI, Professor Kit Parker, and my mentor, Yunsa Lee. And a huge thank you to the Astronaut Scholarship Foundation for this amazing opportunity. This has been such an incredible weekend for all of us, so I really appreciate the chance to be here.
Thank you. Thank you, Jane. We have a couple of questions right there.
Yeah, so my research is not in biology, but it's rather in aerospace engineering, but we also apply particle image velocimetry to measure the velocity of flow fields. So I was wondering, with our PIV, we normally use a ginormous laser, but that would probably melt the heart. So I was wondering what particles you use and what light source you use for your PIV.
So ours is honestly probably along a similar wavelength. So you're using these just really tiny, like green fluorescent particles and a microscope. So I'd imagine it's very similar to what you're doing, but just on a scale that our cardiac tissues can actually handle. But yeah, it's still the same thing, still a laser.
One more question for Jane.
Hi, so I wanted to ask about the actual application of this. Is this for like eventual like surgical or like medical repair, or is this to learn more about the structure of the heart? Or
That's a really good question. So the eventual goal someday with this is to be able to replace an entire human heart, right? So we're starting with just the ventricle. As you can imagine, this takes a while. Like they've been working on this for decades at this point. But we're trying to culture it under those pulsatile conditions so it's more similar to how the human heart actually grows. That way we can create stronger ventricles, right? So that way if we were to use this in a human person, for example, that way it's not incredibly weak. That way we can create like stronger ventricles that would be ready for implantation someday. So that's kind of the general goal.
All right, thank you so much, Jane.
Thank you.
All right, our next speaker is Al-Harith Dame. I hope I said that right. Welcome. And we're going to be talking about executive function development, and please coach me afterwards because that's my job. Welcome.
Well, hi everyone. My name is Hadith. I am a rising senior at the University of Minnesota, majoring in neuroscience and developmental psychology. And I'd like to talk to you today about something I think about a lot and is a pretty cool way of thinking, that being executive function. So, that's the laser pointer. Okay. But before that, I want to talk about you. So, as you know, we as people grow, and so does our brain. So, as we grow, there's an important skill that we develop called higher order cognition that develops with age. And when we say higher order, that refers to the skills beyond the ones that are, I suppose, more basic, but still fundamental, ones that we need to see, feel, hear, or breathe. But rather, these are the skills that are like advanced thinking, reasoning, and planning, which you can really think about as the things that make us human. And one key aspect of this higher order cognition is executive function, which you can conceptualize as a set of attention regulation skills that allow us to control our thoughts, emotions, and actions to achieve our goals. In the context of psychology, you've probably heard of things like intelligence as an important predictor of success. But executive function is different because it's not just about what you know, like it is with intelligence, but rather, can I actually apply what I know, even when it's tough, and actually put that knowledge into practice? making executive function critical. And if you don't believe me still, executive function is really important. I'm going to tell you a bit later about the 3 different skills it encompasses, those being working memory, inhibitory control, and cognitive flexibility. But if you don't believe me, these skills really do matter. Studies show that executive functioning as measured in childhood predicts a variety of outcomes in adulthood, even at age 32. These things include things like health, drug use, criminal convictions, educational attainment, and more. And importantly, executive function challenges are also present in various mental health or neurodevelopmental differences, such as autism and ADHD, making it an important area of interest to researchers in cognitive development. We also know that there are different ways we can measure executive function. And to walk you guys through it, I'm going to show you some games that we actually administer to kids, on an iPad even, as a way of testing these things. So the first thing I'd like to talk about is working memory, or our ability to maintain our attention on one thing and keep it in our heads and manipulate that information as well. So on certain measures on the NIH Toolbox, National Institute of Health, we might ask people to memorize a list of items, such as an elephant, I think that's a mouse, and a cat, and then manipulate them along different dimensions. For example, asking a child, can you tell me these in order of size? Or even mixing them up with another list, saying, I want you to tell me the food first in this list, and then tell me the animals in size order from smallest to biggest. Again, really hitting at, can we remember things, and can we manipulate them and how we think about them? Next, there's inhibitory control, or our ability to selectively focus on one thing at a time and ignore things that are distracting us from solving our plans. So in a task like this, which is called the flanker task, you're asked to pay attention to the way one thing is pointing, for example, this middle fish, while ignoring its distracting members of its school who are pointing the opposite way. Again, can we focus? And finally, my favorite is cognitive flexibility, which refers to our ability to flexibly switch in how we think and see if we can apply new sets of rules to new sets of problems. And I'm going to make you guys do this one. So let's say we want to sort these objects along a dimension such as shape. So if we're sorting by shape, which one do we pick? The truck, that's right. Good job, guys. Okay. Yeah. So then we'll have kids do these like repeatedly back to back, like they're getting super into it, and then we're like, okay, hold up. Actually, I want you to sort these along a different way. Let's do it by color. So if we sort by color then, well, then we're picking the ball. So it gets a little bit challenging, but it's still an important measure of how we can think flexibly. And so we can gauge performance on tasks like these, measuring speed and accuracy. And again, the NIH has developed a set of measures that make it really easy to measure these things, even just on an iPad, making it a really good thing to study. And we also know that these changes co-occur with changes in the brain, namely that of prefrontal cortex development. We measure these using functional magnetic resonance imaging, which measures blood oxygenation, or how much energy our brain is using as a proxy for brain activity. But there are gaps in this research. Namely, these studies often have low sample size due to how time-intensive it is to actually get an MRI scanner to scan people, and because of how expensive it is. I'm talking on the order of $500 an hour, making it really hard to measure these things. But there are solutions. The Adolescent Brain and Cognitive Development Study is a nationwide study following 12,000 kids aged 9 to 10 for up to 10 years, measuring cognition every year and the brain every other, and actually releasing 6-year follow-up data just a couple months ago. And so what I propose and what I want to talk to you all about is my honors thesis, which I'm going to do next year, in which I propose to first characterize executive function development in this data set using a fancy statistical technique that basically predicts trajectories of development, and secondly, examining accompanying changes in brain development by looking at fMRI activation to see how the brain activity changes in different areas of the brain, using another fancy statistical technique that's basically the same thing. And I hope this will help us better understand neurodevelopment, including how these skills develop during this critical time period in adolescence, and also analyze a new wave of data that no one's looked at yet. And so I can't do this without all of you, though. I wanted to thank my 2 labs, the Developmental Social Cognitive Neuroscience Lab and the Developmental Cognition and Neuroimaging Lab at the University of Minnesota, as well as the Astronaut Scholarship Foundation for their continued support. Thank you, and I'm happy to take any questions.
Thank you. We have time for 2 questions. Who's got the first one? Oh, come on. There we go.
What? I'm curious, in your studies or in furthering your studies, when you're doing the sorting, are you using only shapes or do you use words? As an adult, the testing uses word and color.
Yeah, that's a good question. I'll say that this is a more simplified version of the task that we would administer to children, but it can get more complicated in ways you're saying. So, for example, if I can get to it, I put too many transitions in here. Oh my gosh.
Not doing
okay.
this again.
Yes,
Okay,
but the point, okay, there we go. So the point being, we can make it even harder. For example, let's say we're gonna look at the truck, and then we tell the participant, okay, but if you see this word on the side, or if you see this border around the truck, then you're gonna sort it this way. So basically just adding more sets of rules and more complexity in order to get at that, kind of like what you're saying, which we often do in, say, adults. Great question.
Good. One more question, please.
So for your applications of this study, will it be used in like educational like pursuits, like for younger children, maybe those kids with ADHD, like teaching styles, like will it be applied in like school systems?
Yes, that's definitely the goal for this type of thing. Education, as you know, with these neurodevelopmental conditions, those can often present challenges in the educational space. And so the hope is by A, having teachers be more mindful of these challenges people sometimes face, and B, adapting their instruction in order to meet these unique needs, is the hope that eventually we can make a more equitable system of education for people whose brains maybe are just a little bit different.
Good. Thank you so much, Harith. And again, Minnesota, Sunday dinner. Well, as we welcomed Avery Love as our first presenter in this technical conference session 1, now we're going to welcome our last presenter, Grace Coe. She's going to talk about high altitude hypoxia. Welcome, Grace.
Thank you. Hello, everyone. My name is Grace Coe. I am a senior at Miami University, and this research was with the Broad Institute of MIT and Harvard in the world of human genomics. So, did you know that 99.9% of the human genome is identical? The remaining 0.1% is what makes all of us different. It shapes all of our heritable traits, such as your eye color, whether you're lactose intolerant, or if you have higher risk for heart disease. Genomic studies have grown explosively in the last several decades, but have remained vastly Eurocentric. This chart here shows individuals in genomic studies in millions, colored by the population that they're from throughout time. We can see an alarming amount of red representing European populations, and this can be really problematic when we're trying to translate these studies outside of European populations. But African populations, you can see the tiny sliver of purple down there, harbor the greatest amount of human genetic diversity, but are vastly underrepresented. They also carry unique adaptations, including to high-altitude hypoxia. So living at high altitudes poses a lot of physiological challenges due to the reduced oxygen levels. This can lead to developing hypoxia, which can induce responses like high hemoglobin concentration and low blood oxygen levels. And this is really dangerous for expecting mothers and fetal health because it can lead to a lot of pregnancy complications. But Tibetans and Ethiopians have both adapted to these conditions and they're protected from these pregnancy complications. Even though both of these populations have adapted to these conditions, they have adapted in different ways genetically. And while Tibetan populations have been very well studied to the point where we now know the specific gene that is responsible for this, Ethiopians have not been very well studied at all. So, our project aims to understand the genetic variants that impact Ethiopians' adaptation to high-altitude hypoxia and how that impacts reproductive fitness. So, to answer this question, our approach had 2 main steps. Our first step was to identify variants that have recently emerged in Ethiopian populations because this adaptation is relatively recent. And next, we want to identify variants that are associated with reproductive health traits, such as preeclampsia and low birth weight. And then by intersecting these 2 sets, that can help us pinpoint the specific variants that are helping to drive this adaptation in Ethiopians. So in order to do this, we leveraged genomic data from NeuroGap, which is an existing study, and a huge collaboration between the Broad, Harvard, and African universities. We used study site locations to infer geographical coordinates to calculate elevations, which led to classifying around 9,000 Ethiopians as highlanders and around 11,000 East Africans as lowlanders, for comparison. Using this data, we want to find locations in the genome where there's positive selection. So you see a lot of lines here, and that represents an individual. And there's one individual at the top that has a red advantageous variant. And after many, many generations, this variant spreads to the rest of the population, which is positive selection. So in order to find locations like this, we used a tool called Swiffer, which basically analyzes various statistics using that comparison between the Ethiopian highlanders and the East African lowlanders. And then we want to verify that these locations are actually hypoxia related, which will result in a list of positive selection locations. So our preliminary Swiffer results show annotated genes with high probabilities of positive selection. So on the x-axis here, you can see the chromosome location, and the y-axis shows the probability of positive selection. We actually found a lot of genes that were uncharacterized, meaning we don't really know their function. But this is really exciting because it's showing diversity in action, and these are genes that have likely not have been studied very well before. But we also found the ADH1C gene, which has been previously reported to have been positively selected in Ethiopians, which serves as a nice positive control. Next, to assess reproductive fitness among high-altitude Ethiopians, we analyzed existing genome-wide association studies, or GWAS, on traits like birth weight, preeclampsia, and gestational diabetes. GWAS essentially examines the whole genome to find variants that are linked to specific traits. So this is a cartoon representation of it, where we see the chromosome on the x-axis and the significance level on the y-axis. So the taller the bar, the more likely this variant is associated with this reproductive outcome. We will then overlap the reproductive trait GWAS results with the positive selection loci we found earlier to identify Ethiopian-specific variants that are driving this adaptation. After this, we will conduct downstream enrichment analysis to link the genes to specific molecules that are helping Ethiopians adapt to hypoxia, and hopefully this can help us advance our understanding of hypoxia resilience and inform better therapeutic targets to mitigate hypoxia-induced birth complications. And before I close, I'd love to thank the Martin Lab at the Broad Institute, as well as the Lee Lab at Miami University, and of course the Astronaut Scholarship Foundation for this wonderful opportunity and support. And thank you so much. I'll take any questions.
Thank you. Thank you, Grace. We have a question for Grace. There's a question. Where's my microphone? Well, I have my microphone, but you know what I mean.
Hey, so I was curious if these people have adapted to these challenges from having low oxygen. Is there any investigation into if those people move from an area with very low oxygen to like the lowlands with a more standard amount of oxygen? Do they have different effects? Or do they have even better outcomes? Or has anyone looked at that?
Yeah, so we're actually not really sure about populations that have lived in higher altitudes and moved to lower ones, because Ethiopians themselves, they have migrated to higher altitudes maybe 40,000 years ago, but their adaptations actually came in maybe 5,000 years ago. So this is relatively recent compared to when they first migrated. So this is really exciting just to see get all this data from Ethiopians, because it's also the first time that such a large amount of data has been collected from these populations to study that. But I'd be interested to see as well. Yeah, thank you for your question.
Thank you.
Yeah.
One more question for Grace.
I was just curious. You didn't mention in your program the altitudes at which the Tibetans and the Ethiopians were. I would like to know what the altitudes were and if there was any difference in altitude.
Yes.
Thank you.
Yeah, great question. So high altitude is technically classified as anything over 2,500 meters, or that's around 8,000 feet. But I think, to my understanding, Tibetans live at a higher altitude compared to Ethiopians, but they're both well over 2,500 feet, so we classify them both as highlanders. Thank you for your question.
Thank you very much, Grace. Thanks for closing us out. Wow, before I say the incredible round of thank yous that need to be, I have a public service announcement. So our scholars in group 2 with a star on the back of your name tag should immediately head when the session is over to the speed learning at 11:30. You have about 10 minutes. So scholars in group 2 go to speed learning at 11:30. Scholars in group 1 go at 1:30. Are we good? 2 at 11:30, 1 at 1:30. Yes? Do I hear an amen? Thank you. So before we, again, we're right on time. Woohoo! So first of all, I want to absolutely thank all the scholars that presented to us this morning. Incredible, engaging, thought-provoking. Wow. Thank you all. I'm going to have thoughts about Ethiopian heights and killing things and all kinds of nonsense. Because I'm on steroids, and that'll just fuel that fire. All right. At this time, again, we want to thank our scholars for being here, and you know what your next activities are. There's the Professional Expo right outside, which is fantastic. Please take advantage of those conversations and that networking capability. Also, for your speed learning, we just talked about where's group 1 going and when? Okay, great. And where's group 2 going? Same place. Good, excellent. Don't forget lunch. We do need to eat. And also make sure you've checked in for all of our guests using the QR code. And we will resume this fantastic technical conference at 3 p.m. Central Daylight Time in this very room. So go enjoy this beautiful hotel, enjoy networking, talking with one another. And again, thank you to ASF and thank you to our incredible scholars and all of you who support them. Have a good rest of the day, and we'll see you at 3.